Thank you so much, all of you. We have with us today Dr. Praveena Bhandari. Ma'am is Associate Professor, Department of Biochemistry, Shri M.N. Virani Science College. She is a very experienced faculty member, and she's been with us for 28 years in academics. And she's going to take plant hormones, which is one of the very essential topic that is covered in most of your examinations. I hope you take the best out of her. And last few minutes will be dedicated to questions if you have any, or you can drop them into the group that you're already joined in. So I request ma'am to kindly take the lecture. Thank you.
Sharing. Yes, good morning. Am I audible to all of you? Fine. So today we'll be discussing about plant hormones. You all must be aware about plant growth. It is irreversible, that is, once the plant will grow, it will not revert back. Permanent, once the seeds germinate, it will grow and remain permanent there. That is, during the plant growth, there is increase in the size of entire plant, organ, tissue, or cell, and it also involves metabolism. So that leads to plant growth. Who governs these processes? There are various factors: genetic factors, external environmental factors, and also all these factors are governed by chemical hormones. So today we'll be discussing on phytohormones, hormones found in the plants. They are different from animal hormones. Mainly five plant hormones are known. They divided into two parts: growth promoters and growth inhibitors. Growth promoter hormones are indole acetic acid, also known as auxin, gibberellic acid, cytokinin, and growth inhibitors, or also known as retardants, are abscisic acid and ethylene. Now today I want to tell you, share something very interesting with you. Discovery of all these hormones was accidental, not just they were finding something, but just because of the observation. Today's my lecture's key word is observation. You will be surprised to see that how this observation technique has led to so much of discovery. The plant hormones, hormone word itself means in Greek to excite. They are always very active in small amounts. They are produced in one part of the plant and have the function in the different part, transported to another for their action, and most important, their action is specific for that site. Not that any hormone can do any function. They cause physiological or developmental responses which can be stimulatory or inhibitory. As I told you, these plant hormones are different from animal hormones. Animal hormones, they are made in specialized tissues, endocrine glands, you must have seen somewhere. And these animal hormones have specific target. They will go to only that particular tissue and so the effect. Whereas in the plant hormones, they are also made in specialized tissue, but they do not have definite target areas. They cover lot of area in the plant. For example, auxins can stimulate adventitious root development in a cut shoot, or they can also cause shoot elongation or apical dominance. Today we'll be discussing about their discovery, chemistry, function and applications. And another important interesting thing about the hormones are they have multiplicity of effect. That means their effect will depend on the site of action, on the developmental stage of plant, whether the plant is in the stage of a seed or fully grown plant or it is a bird, and also the effect depends on concentration of hormone. In our today's class we will discuss how the concentration also affect the effect of that hormone. This we have already seen. Now coming to the first hormone, auxin. Auxin is a Greek word which means to grow, and chemically it is indole 3 acetic acid. Again, I said observation. Years back in 19th century, Darwin, you will be coming to those names, just a plant was growing near a window. They saw that the plant bends towards the light. Why this happened, how it happened, that is lead to discovery of auxin. So it was seen that first, Charles Darwin and Francis Darwin, his son, in 1880, they observed that the canary grass coleoptile bent towards light. If you cut the apex and put the light, illuminate that plant coleoptile, it is not showing the movement. Later on, in 1913, Boyson and Jensen, they did one experiment using mica plate. What they did, as I told you, how simple observation can lead to great discovery. In this experiment, they put mica plate on one side of the plant, of the tip coleoptile, and illuminated that part. You can see the red arrow here. They saw that the part which was dark, not illuminated, it bent. In the second experiment, they inserted this and again eliminated it. The plant was inserted. It showed a bend. Why this happens, how this happens, this was exactly discovered and isolated by scientist Wendt. So as soon as we hear the word auxin, the terms come in our mind. Scientist Wendt in 1926. What he did? Again, observation. He took the coleoptile of Avena plant. He decapitated, removed the cap of the coleoptile and put on a agar block. What happened? They observed something. This agar block was kept on the decapitated coleoptile and eliminated from one side. There was bending observed. Why this bending observed? The reason is there is one chemical secreted, synthesized at the tip of the coleoptile. It diffuses downwards, and when the agar block was put, the agar block has this auxin, that chemical, and when this chemical was placed like this, it diffused down and the elongation was seen. One another experiment they saw, what Wendt did, he decapitated it and then put the light. The light, there was no bending. No bending because auxin was not diffused down. Here this can be seen. It was decapitated and you eliminate, but because of the absence of the cap responsible for synthesis of auxin was not present, and so bending was not observed. So he discovered, he isolated, and also the term auxin was given by this scientist Wendt. Now coming to the this we saw, and by this experiment it was seen that if you want to grow anything, bend one part. That means the cells should grow from that side, then only the tip can bend. This can be very well understood by the example of parade, row of 3. At the corners, if you want to see them straight, the last person in the corner walks fast. Same way, these cells elongate more. So Two conclusions were proved by this experiment. Auxin diffuses downward and it is more effective in the darker side here. The chemistry part of auxin, they are derivatives of indole compounds. Natural auxins are formed from the amino acid tryptophan. So, amino acid tryptophan serves as the precursor for natural auxins. The natural auxins are two known as indole acetic acid, also abbreviated as IAA, and indole butyric acid, IBA. Besides these uh, auxins found in the plants, we also can prepare them artificially, known as synthetic auxins. Three of them I have listed, important ones. First is naphthalene acetic acid, NAA. Second one, 2,4-dichlorophenoxyacetic acid, also known as 2,4-D, and similar 2,4,5-T, that is 2,4,5 trichlorophenoxyacetic acid. Now these three are the synthetic auxins. The auxin can found in the free forms, or also they are available in the bound, which can be isolated using organic solvents. There are some substances which stop or inhibit the action of auxin, known as antioxins. The site of production is the shoot and root tips, that is also known as the apical meristems. Transport is in two ways, basipetal and acropetal. Downward movement is known in the shoots is known as basipetal movement, and in the roots it is upward direction known as acropetal. And this movement controls the overall shape of the plant and helps the primary stem to be the leader in the plant. Functions of auxins. The main part, it promotes cell division, initiates lateral root formation. Third function, it causes apical dominance. The lateral bud is not allowed to develop till the apical bud is present. It promotes flowering in the pineapple special plant, induces parthenocarpy, that is seedless fruits in tomatoes. It prevents premature leaf fall and fruit fall. So, based on these functions, it has got various applications. First is it promotes cell division. So, based on this, it can be used in tissue culture. Many of you would be familiar about plant tissue culture experiments. It is used to obtain plant from the stem cuttings because it initiates lateral root formation. Then it as a synthetic auxins will kill dicots, so it is also synthetic auxins are used as weedicides. Then apical dominance will also help in bonsai making and hedges. If you remember the gardens having the hedges, because the late apical buds are removed, so the plant will grow in the lateral direction. As promotes flowering, some of the important plant is pineapple. So more fruits, more money can be made. Induces seedless fruits and vegetables, very important to have grapes without the seed. You can enjoy the fruit eating without seed. It prevents premature leaf and fruit fall, so increases productivity. This is the example of apical dominance. The apical bud is dominant, the lateral buds are dormant. This elongation mechanism is this auxin will stimulate pumping of H plus. This H plus activates the enzyme which are responsible for the tight bondaging of the cellulose molecule. So these bonds are broken. As a result, cellulose loosens and the cell wall elongates, and then water uptake is more. This is the mechanism for elongation of cells. Next hormone is gibberellin, derivatives of terpenes. The story behind this is in Japan. few farmers, they observed that the rice, the common rice plants are around 3 to 4.5 feet, but some plants were exceptionally tall. Some rice seedlings, they became very very tall, around 12 to 15 feet, and this in Japanese they called as Becken disease. Why? Because this 12 to 15 feet plant, the stem is very weak, they all started falling down. So that was a foolish behavior. So foolish behavior is known as Becken, and based on this, this were named as foolish seedlings. This was observation by farmers. Now scientist Kurosawa, he found that these rice seedlings were infected by a fungus named Gibberella fugiculae. And so, the active substance responsible for this foolish behavior, tall growth, they were named as gibberellin and later known as gibberellic acid. Around 100 gibberellic acid have been isolated, but most studied and the first isolated is Ga3. The site of production is plants, seeds, and young shoots. Their function is, more important function is, they control cell division and elongation, but along with auxin, the transport carried by xylem and phloem. The functions of gibberellic acid: it promotes stem elongation, and because of this we will see three applications. It promotes malting process in fermentation studies, delays senescence. Senescence is aging in the plants. The plant becomes old. Increases seed maturity. Bolting, that is, what is bolting? Here you have, this is a rosette plant. Rosette plants are plants in which the distance between the two leaves is very small. Example is cabbage. So what is bolting effect? Bolting effect is induced by gibberellic acid, and these, another example is beet. So the leaves are very close. So when you spray gibberellic acid on it, one leaf will come from here, second leaf from here. So internodal distance increases. As a result, it causes elongation, and this effect is known as bolting. So let us see how it will be useful. As I told you, the gibberellic acid promotes stem elongation. So this will lead to elongation of grape axis. So you will get good, fresh, juicy fruit without any damage. We'll see that. I'll show you the picture in the next slide. It increases the height of sugar cane. So if you remember, in the sugar cane, the middle part is responsible for formation of sucrose. So more height, more sucrose, more money. Elongation of apple fruit. Here the diagram. The shape of the apple fruit can be made larger and the fruit also can be made bigger. The malting process is used in the brewing industry. Then delay senescence means, what happens if you spray gibberellic acid, the fruits can be left on the plants longer. It will not cause the plant to age fast. So the farmer can sold its fruit in batches. Instead, what will happen if you do not use gibberellic acid? All the fruits in the farm will ripe at the same time. But because of this gibberellic acid, the senescence can be delayed. Another important function, it promotes seed germination. Dwarf seed, this is the effect of gibberellin. The internode distance is increased. So long plants. About the grapes, what I was trying to tell you, this internodal axis is elongated, so the fruit gets enough space to develop and increase in the size also. You will have all good quality fruits. Third hormone is cytokinin. It is believed to enhance cell division. The story of this is two scientists, Skoog and Miller. I told you this whole story is based on observation. Skoog and Miller, they observed that cells of the internodal region of the stem of tobacco plant, they showed proliferation, increase in the size, only if auxins were accompanied with some other substance. And this substance was isolated from herring sperm DNA, and this was called kinetin. Okay. They used extracts of vascular tissue, coconut milk, yeast extract, DNA extract, and later on, this substance was found to be cytokinin, helping in cell division. Cyto is cell, kinin is division. Chemically, it is 6 furfuryl amino purine. They are purine derivatives formed from adenine, and these cytokinins work with some other phytohormone. So, synergistic effect. We'll see to it very soon. Role of cytokinin. As some of you would be familiar with tissue culture experiments, we artificially provide all the environment, nutrients, and here the hormones play important role. If the concentration of auxin and cytokinin is same, equal, it will promote callus growth. That is, cell division will be promoted but not cell differentiation. That leads to callus growth. As the plant starts developing, the concentration is regulated. If the auxin concentration is more than cytokinin, the cells will help differentiate and start forming roots. Reverse is true. If cytokinin concentration is high than auxin, the differentiation will lead to shoot formation. So we can see that when the concentration of two hormones, auxin is equal to cytokinin, it will lead to only cell division. But if one is higher, it leads to cell differentiation. Very important concept, asked often in your entrance test, vivas. Other facts about cytokinin. It has a molecular structure similar to adenine, naturally occurring zeatin, isolated first from conziame, scientific name, is the most active of all cytokinins. Cytokinins are found in root tips, seeds, fruits, and leaves. These are the areas where maximum differentiation will occur and cells will divide in different uh, parts of the plant. They are transported in the xylem and work in the presence of auxin to promote cell division that we saw in the first hormone. Functions of cytokinin. As we saw, it promotes cell division, fine, but also it promotes cell enlargement and cell differentiation. It promotes adventitious root development, leaf formation, and enlargement. It breaks the seed dormancy by increasing the metabolic activities. The metabolic reactions, the enzymes responsible for them, are formed more, and as a result, the seed dormancy is broken. It delays senescence. What is senescence? Senescence means aging process. So one experiment was done by Richmond Lang. He took two leaves. One leaf was dipped in cytokinin solution, other was just in water. It was seen that the leaf dipped in cytokinin delayed aging process. That means it stayed younger, or younger means green for longer time. So that is known as Richmond Lang effect. That is delaying of senescence by using cytokinin. It promotes flowering in some plants. It also helps in promoting secondary growth in dicots. Secondary growth is the formation of cambium, you know. So there is the in between the cambium, the layer is formed, and that is favored by cytokinin. Next hormone is ethylene. The minute you hear about ethylene, first thing comes in your mind is it is a gaseous phytohormone, the only hormone available in gas form. And the most simple structure, ethylene. You must have heard of the saying, one bad apple spoils the barrel. What is the basis for that? If you keep a really ripe brown banana, if it is kept right next to a bunch of green are the unripe bananas, they all will ripen and turn yellow much faster. What has happened? Actually, the brown banana is communicating with the green bananas using a hormone called ethylene. That ethylene hormone is released from the brown ripe banana, and it will affect the ripening and rotting of the plants. So that is the reason why it said one bad apple spoils the barrel. Barrel is the full of apple, and this has the basis on its effect of ethylene gas. This also has its story. It IUPAC name is ethene, double bond C, double bond C. It is a gaseous hormone produced by the breakdown of methionine in the plants, and the site is the cells undergoing senescence, that is the aging process, and the ripening fruits. They will produce methionine, and that will be broken down, and that will lead to formation of the hormone, gaseous hormone ethylene. Also, what we use in our horticulture, it is the liquid form, and liquid form is known as ethiphon. About this hormone, two scientists are well known. One is Sarah Dowd. He discovered the role of ethylene in senescence. Another scientist, Crocker, he discovered the role of ethylene in ripening fruits. So, two important roles discovered by two separate scientists. Functions of ethylene. As we already saw, two most important functions we have coming to the inhibitory hormones, growth inhibitors. It will cause plant aging in plants, that is senescence. It promotes ripening of the fruits. The fruits will ripen fast, as I told you about the banana and the apple. It helps in increasing length of petiole and internode in the deep water rice plants, that is in the, so it will help there in the seedlings. It breaks seed dormancy. How? It will cause the seed to get germinate, so it will promote seed germination, specifically seen in the groundnut seeds. It promotes sprouting in potato tubers. You must have seen, potatoes with eyes, and that is because of sprouting. So it is helpful, done by ethylene. Ripening part of the fruit, if it is placed near the unripe, it also gets ripened, already discussed. It promotes flowering and flower setting in pineapple. And the last, ethephon, aqueous solutions used for ripening of the fruits artificially. About ethephon, I would like to say that it what happens is, this is aqueous solution. When we use it, ethylene gas is released, and that is used for ripening of the fruits. Here I would like you must have seen the bananas what are available these days. What happens is, if we do not go for artificially ripening, the fruit becomes overripe. So most of the farmers are preferring to do this artificial ripening. And in this, what happens, the artificial ripening is from outside to inside. You must have observed the banana peel is yellow, but when you peel it, the banana is unripe, not very raw, very ripe. So, artificial ripening is always from outside to inside, and the naturally ripe fruits will be from inside to outside. Another important use of this ethephon is, it causes thinning of cotton fibers, which is very helpful for commercial use. Another important, it causes thinning of walnut shell. So walnuts can be easily broken out and taken out. It promotes formation of female flowers in cucumber in specific plants, not everyone. Ethylene also promotes flowering and flower setting in pineapple. So, what happens? We discussed that three hormones, auxins, cytokinins, and gibberellin are plant growth promoters, and two are inhibitors, ethylene and abscisic acid. But what we discussed about ethylene, this hormone has both the categories. It is promoting also and inhibiting also. So this hormone comes in between the two. It promotes certain activities and it retards aging process and some other processes. Now, because of this ethylene, farmers can do smart business. What is this smart business? As I told you, the unripe fruits can be taken from one part of the country to another part and do business, and then once they reach the site of consuming, there the pressure is maintained in such a way that then the ripening process starts. So this formation of ethylene requires oxygen, and the industry is using this concept. So by controlling the partial pressure of oxygen and carbon dioxide in the truck which is used in the transport, the ethylene synthesis can be prevented, and thus we can slow down the ripening process. So what happens, the farmers, they don't want their products to go bad before the consumer buys it. So by this, by use of ethylene, we can do the farmers can do smart business. And the last hormone is abscisic acid. The spelling is bit confusing. Try to remember scissor spelling and then we can write the name abscisic acid. Here the story is three independent researchers found three plant inhibitors and they individually named it as inhibitor B, one group, second group named it as abscission 2, and third group named it as dormin because they were inhibiting the plant growth. Later on, they found that these three were chemically identical because three different parts of the world they did this experiment and they all were found to be abscisic acid. So all those growth inhibitors, which are inhibiting the plant growth, they are named as growth inhibitors. Isolation of this was done by Liu and Kranz first time from mature cotton fruits. So they were the one who got the credit to isolate it. Observation is fine, but you have to go about isolation also. Sorry. Now, as we know, it is a general growth inhibitor. Structure is similar to gibberellins. The site of abscisic acid production is again all organs, roots, leaves, stems, and fruit. All parts of the plant produce abscisic acid. The action is counter of what plant growth promoters are doing, auxins and gibberellins. As we saw, auxins and gibberellins are responsible for having plant growth promoting effect, but this abscisic acid will lead to some inhibition, and so it is called as having counter effect antagonist. What it does, it maintains dormancy in the seeds and buds. Seeds will not germinate, they will prevent it from germination, the birds will not be developing. And also another important function which you always remember, as soon as you hear of abscisic acid, it will close stomata. Why stomata will be closed? To conserve water. And what it will do, I'll show you in the next two slides how it will stimulate guard cells. So, this hormone is always produced in response to stress. These days, stress is very common to human beings also, but we are quite intelligent, quite efficient to manage the stress. Same way, the plants are managing the stress by producing abscisic acid. So, as I told you, it is a growth inhibitor, yet not proved. Direct role is not proved. So, what is believed is abscisic acid lowers the process of RNA translation. You must be well aware that gene expression involves two processes, transcription and translation, and then the proteins are formed. So this abscisic acid decreases or lowers the process of RNA translation. As a result, important proteins are not formed, especially amylase and proteases, which are promoted by gibberellins to have the positive effect. But how abscisic acid is reducing activity is not known. But this abscisic acid does not let germination occur. And as we are well aware that gibberellins having germinating effect, it promotes synthesis of amylases and proteases. But because of the presence of abscisic acid, the germination will not occur. And why it will not occur? Because the amylases and proteases enzymes required to break the starch and proteins respectively from cotyledons, these processes are lowered or stopped, but the direct role is not proved. So coming to the functions of abscisic acid, as it is having functions opposite to what we discussed of auxin, gibberellin, cytokinin, it is known as a antagonist. It promotes or initiates senescence, that is aging. Yes. Second function, it promotes or initiate abscission. Abscission is what? How it happens? You can see this diagram I want to show. Here is the leaf. Petiole is there. At the end part here, at the bottom of the petiole, junction of the attachment, here one abscission zone is formed of suberin. This abscission zone will block the transport or exchange of gases and water between the leaf and the stem. As a result, it will cause senescence or it will cause abscission. The plant leaf will start fall. It promotes dormancy in the seeds. Dormancy in the seeds means the even if you supply favorable conditions, the seed will not germinate. And also it is useful in the extreme dry conditions. The seeds can withstand the extreme dry conditions. It inhibits germination, as we saw just in the previous slide. It inhibits the production of amylase proteases. So antagonist action to gibberellic acid. Stomatal closure. In the extreme dryness, two conditions are there, whether the temperature is very high or the water availability is less. In both the conditions, the plant would not allow its water to evaporate. So we need to close the stomata. And how it is done, I'll just show you in the next slide. Once we finish the last function, the last function is it promotes bird dormancy. That is, the bird will be not allowed to germinate. And the main important role looking at all these functions is it helps the plant to survive in the adverse conditions, in the stressed situations. So it is also known as stress hormone. The mechanism of abscisic acid, how it works. If you see this diagram, these are the guard cells, stomata or stoma, and these are the epidermal cells. So what happens in the dry condition, extreme conditions, abscisic acid travel to the guard cells, sending a message that water is low, availability of water. So this will cause the guard cells to rush the charged particles K plus. They will leave the guard cells and come in the epidermal cells. When K plus leaves, the water will also go from the guard cells to the epidermal cells. As a result, the guard cells will shrink. Shrinking will cause this space to come closer, and as a result, the stoma will close and the water will not be able to exit through the stomata. So, this is the mechanism of stomata closure done by abscisic acid. So, abscisic acid is also known as the thirst signal. The minute the plant is thirsty, water is not available, high temperature, it will alert the rest of the plant that it is water stressed. So many things will happen. Now, to summarize the entire class, what we discussed in this session: 5 hormones, 3 are growth promoters: auxins, also known as indole acetic acid; gibberellins, gibberellic acid also they are known as; and third is cytokinin. They all three are growth promoters. Auxins main function cell elongation. Gibberellins, they also cause cell division along and growth along with cell elongation. And cytokinins are mainly involved in cell division. Plus, they also inhibit senescence. 2 are growth inhibitors or retardants, abscisic acid and ethylene. They cause abscission of the leaves and fruits. The younger leaves will not fall, but the older leaves fall, how we just now saw, suberin in the abscission zone. And they also cause dormancy of the buds and seeds. Ethylene, as we saw, it promotes senescence and fruit ripening. Now, one message for all of us: have to observe people around and identify who are our growth promoters and growth inhibitors. Here I mean your personal growth, and you also should find out who are having synergistic effect and who are having antagonist effect. Something more I would like to share: we are really good at judging and observing others people, but not myself. And this corona pandemic has taught us to go inside, and this will lead us to become genius. How? Last slide. Key to become genius is, all genius is simple. What we have to do is close observation of the self and have self trust. And the last sentence is, a talent does what he can, a genius does what he must. So I wish you all the best, and thank you for listening to me, and wish you all the best to be a very good observer and to come out with very colorful colors and achieve a lot in your life. Thank you. Yes, Saminarayan. Yes, please unmute your mic and ask me. Okay. You can write in the chat box. Okay, they can. Yes, your question. I think you don't have right for unmuting. You can write in the chat box. Okay. You are getting rights to get unmuted for 2 minutes. Keep your question ready.
Uh, you have been assigned rights to unmute yourself and ask questions. If you have any question, please unmute yourself and ask. Uh, I think ma'am will take up the questions in the chat box later. I don't think they are willing to ask it right now. Thank you so much, ma'am, for your wonderful presentation and your motivation. Dear students, our next speaker for the day is Dr. Shweta Bhatt. Ma'am is presently working as Assistant Professor, Department of Biotechnology, Atmiya University. Ma'am has a teaching experience for more than 12 years and she is an expert in molecular biology, genetics, and recombinant DNA technology. She'll be taking the topic today, which is very important for competitive point of view, that is signal transduction. I request ma'am to kindly start with her lecture. Shweta ma'am, I've assigned the rights to you.
Okay, ma'am. Thank you. Am I audible? Ragini?
Yes, ma'am. Yes, ma'am, you're audible.
Okay, done. So let us start. Good afternoon, everyone. Let us learn one more important topic for your competitive examinations, that is signal transduction. Okay. So first, let us start. What is signal transduction? The overall process of converting extracellular signals into intracellular response, including large number of steps in between, is called signal transduction. To make it more clear, let us look through the figure. It says, what is signal transduction? When a signal coming to the cell and there are number of steps going inside, and at the end the cells shows some type of response, the whole process is called as signal transduction. When I say a signal coming to the cell and the cell has steps and the response, then what type of responses? So there can be large number of responses which a cell can show. For example, activation of enzyme activity, changes in the ion permeability, activation of DNA synthesis and RNA synthesis. These all are the type of cell responses which a cell shows when a signal binds. Or it can be modification of cellular metabolism and gene expressions. Now, when I say about signal transduction, the word first comes is signal. So let us see what is signal. It can be called as signal or ligand or signaling molecule. So what do you mean by signal? Signal, these are the molecules which are released by signal producing cell. So these are the signals, the molecules which are produced by one type of cell which is called as signal producing cell, which then transfers the information, that signal, to a particular target cell so that the other cell can start the response. When I say signal, then the other partner comes called as receptors. So receptors are the specific membrane proteins which are able to recognize and bind to corresponding signal or ligand or signaling molecule, and the receptor becomes activated and it transfers the signal inside the cell. So this is receptor. When I say receptor, what is it? Receptor is basically present on the target cell to receive, welcome the signal, and then it gets activated, and after it itself get activated, it transfers the signal inside the cell, and then the cascade of reactions will occur. Here one more thing you all would be knowing, but let us have a revision of that. When I say about receptor, it is a transmembrane protein, generally transmembrane, which cross the whole membrane, and they have two parts: hydrophilic part, which is outside, you can see, and the membrane part, which is embedded inside the membrane, it is hydrophobic. So this is hydrophilic part and this one is hydrophobic. Okay. Now the whole signaling process can be divided into many types, but the basic types of signaling are three. So the three basic types are endocrine signaling, paracrine signaling, and autocrine signaling. So what do you mean by endocrine signaling? When I use the word endocrine signaling, it means here we have again one cell would be producing the signal and there would be some target cell. So in endocrine signaling, what happens? Signal producing cell is there, it will produce the signal and it will go distant to a far distance and binds to a receptor on the distant target cell. A target cell is far away. And a very nice example for endocrine signaling is endocrine hormones. So it is very much clear when I say the word endocrine hormones, because you all are knowing endocrine hormones. For example, if I say pituitary gland, you know the location of a pituitary gland. It produces so many different types of hormones like ACTH, like growth, like luteinizing, like follicle stimulating. They all are synthesized by the pituitary, and through blood vessels they move to different parts of the body distant from the pituitary, the signal producing cell. So this is one type of signaling pathway which is called as endocrine signaling. The second type of signaling is called as paracrine signaling. What is paracrine signaling? When the signal producing cell and the target cell, they are near to each other. The signal producing cell and the target cell are near to each other. It means the signal and the receptor for the nearby target cell are close proximity. The example for paracrine signaling is transfer of neurotransmitters in the nerve cells. That is an example of paracrine signaling. Moving ahead, the third type of signaling is autocrine signaling. Auto means self. So what is autocrine signaling? Autocrine signaling says the signal producing cell and the target cell both are same. So here the picture is telling us very well the signal produced by the cell and it goes and binds to the receptor on the same cell. That is called as autocrine signaling, and when the signal binds to this receptor of the same cell, it starts some chain of reactions inside and the cell shows the response. Example for autocrine signaling is cancerous cells. In cancerous cells, the cell itself starts synthesizing large amount of growth factors, And these growth factors, they start releasing and they bind to their own receptors, which gives the signal to the cell to start growing, dividing fast. So that is autocrine signaling. Now, classification of signals. Signal is ligand is signaling molecule, same thing. So if I say about classification of signals, the signals can be classified as many ways, but on the basis of the nature of signal and its location of a receptor where it goes and binds, the signal can be classified as three types. The first one is hydrophobic signals या molecules binds to intracellular receptors. So the first type of signals are hydrophobic in nature, first point, and on the basis of location of receptor, if I say, then they are having intracellular receptors. The examples of hydrophobic molecules going inside the cell and binding to the receptor are steroid hormones. All type of steroid hormones, lipid in nature, so they can cross the membrane very easily and they bind to the receptors inside the cell or the nucleus and basically promotes different type of process like increasing the transcription or DNA replication. These are the different types of steroid hormones. The second type of signals or signaling molecules are hydrophilic signaling molecules or hydrophobic signals which binds to the cell membrane receptors. Means where they will bind to the surface of cell. They will not go inside the cell membrane. Why? Because they are hydrophilic in nature, they cannot cross the hydrophobic layer present in between the plasma membrane. So the examples of these type of signaling molecules are neurotransmitters like acetylcholine, epinephrine, or it can be peptide in nature like insulin hormone, like gastrin, it is also a peptide hormone. So this is second type. Now third type of signaling molecules or signals or ligands are those which are hydrophobic in nature, but still they are hydrophobic. It means they are lipophilic, so they can enter the plasma membrane. But these are those hydrophobic molecules which binds to extracellular receptors on the cell surface, on the cell membrane, not inside. The example for these type of molecules are icosanoids. Maybe you would have heard the name icosanoids. These are basically group of compounds which includes 4 prostaglandins, prostacyclins, thromboxanes and leukotrienes, which plays very important role in vasodilation, constriction and many type of muscular activities. So these are those molecules hydrophobic, but then to bind to extracellular receptors because they get broken down very fast. So they basically are synthesized and binds to the neighboring target cell on the membrane, and then their functions, the receptor starts doing the work. So if one more point I add, so icosanoids basically work in paracrine signaling, I can say. So all the 4 types of icosanoids basically are synthesized from a phospholipid. That can be one more point which you can remember. Next is families of cell surface receptors. Now receptors can be many. Let us discuss about cell surface receptors. Receptors, so now it should be clear, I think. Receptors means I am talking about the transmembrane proteins present on the membrane binds to a signal. So when I say about receptors, cell surface receptors, they are basically of 4 types. First, there are those receptors which has intrinsic enzymatic activity. Second, there are receptors which does not have their own enzymatic activity. Because receptors are protein, they can have some motifs attached, motifs in them which can act as enzyme. It is possible. Third comes those receptors which actually act as ion channels. And the fourth type of receptors are G protein coupled receptors, the most important receptors of all. Let us see one by one. So the first one I said, receptors with intrinsic enzymatic activity. What do you mean by this? This means there are receptors in many cell membranes which are, so this is one example of a receptor, transmembrane protein. So, and this is receptor with intrinsic enzymatic activity. So here you can see the receptor has its own enzyme motif, for example, tyrosine kinase domain. Okay. So receptors with intrinsic enzymatic activity means receptor has enzyme unit attached or inbuilt in itself. The largest family of receptors which have intrinsic enzymatic activity is one example is receptor tyrosine kinases. This is the largest family of the receptors, and these receptor tyrosine kinases binds to various types of signals responsible for cell responses. Few examples are insulin, various growth factors like epidermal growth factor and fibroblast growth factors, and others. If we see this slide, we see that there are two monomer units. Now we are talking about receptor tyrosine kinase, isn't it? So here you can see how the receptor tyrosine kinase works. Now one thing is clear, this is a receptor. Now this is the receptor, okay? And this is the receptor tyrosine kinase which has what? Which has tyrosine kinase activity here in the receptor. But it will not be active until a signal binds. So what happens in receptor tyrosine kinase? The receptor is made up of two subunits and the two subunits are inactive. But the time signal comes and binds, the two monomer units get combined and it leads to activation of the enzyme present here. Which is the enzyme? It is tyrosine kinase. It becomes active. As the name suggests, it is tyrosine kinase. So that would be coming further also. But what is tyrosine kinase doing? As the word kinase is there, it will phosphorylate first itself to get complete activation. And at which amino acid? Tyrosine. And because after this enzyme gets activated, it will now phosphorylate its target protein. Okay, so till now, how many cascades we have dealt about? We dealt a ligand will come and bind. It will leads to the activation of tyrosine kinase enzyme present in the receptor towards cytoplasmic site. When it gets activated, it will phosphorylate itself first for its complete activation, and then it will phosphorylate its target protein. This much you should know about this thing. One more important point about receptor tyrosine kinase is that receptor tyrosine kinase plays a very important role in the RAS-MAP kinase pathway. It is a very big pathway, but what major point you should know for your competitive exam is that receptor tyrosine kinase plays role in this type of pathway where there are two words which you should know, RAS and MAP. So RAS is a GTP protein, GTP binding protein, which basically helps the MAP pathway. What is MAP? It is mitogen activated protein kinases. Mitogen you all are knowing, which plays role in the mitosis. So what is the role of this whole pathway? In regulating the cell growth and cell differentiation. So the RT case, that is receptor tyrosine kinase, plays role in the cell growth and differentiation through a pathway which is called RAS-MAP kinase pathway. RAS, one more point, RAS, why it is called as RAS? Because it was first identified as the oncogenic protein of tumor virus that causes sarcomas in rat. So that is why it is called as RAS. I think you would be knowing. So that is why rat sarcoma virus. So from RA and as the name came to RAS protein. So RAS protein, it is GTP binding protein and it will be active when it is bind with GTP. Okay, it should be clear. Now, the second type of receptors. First we have dealt, that is receptor with intrinsic enzymatic activity. Now, second type of receptors are there present on the cell membrane which do not have intrinsic enzyme activity, not intrinsic enzymatic activity. It means the receptor itself does not has inside any type of enzymatic activity. So then what happens? So for example, if this is a receptor which does not has an intrinsic enzymatic activity, but in that case, the enzyme gets non-covalently attached to the cytoplasmic domain of the receptor. Okay, enzyme will work, but enzyme receptor has not having of its own, but the other enzyme will come and bind non-covalently with the receptor. There are many receptors which does not have intrinsic enzymatic activity. One example you should know, one is cytokine receptors. Cytokine receptor family are those receptors which will bind to cytokine ligand. Very simple. So these are cytokine receptors which will bind to cytokines like interleukin, erythropoietin and others. These type of receptors also binds to some other polypeptide hormones, for example, growth hormone. One more major point which you should know is the main function of cytokine receptor family is if a cytokine binds to a receptor, the major function of these cytokine receptor is like in the earlier RTK, receptor tyrosine kinase, we read one example, that is RAS MAP pathway. Similarly here for cytokine receptors, you can learn JAK-STAT pathway. That cytokine receptors basically leads to the activation of which type of pathway? It is called as JAK-STAT pathway. JAK stands for Janus kinases. JA stands for Janus, K is kinases, and STAT is signal transducers and activators of transcription. So overall, in a summary, you can learn that cytokine receptors basically helps whenever this ligand will bind to the receptor, there will be many reactions inside the cell which will finally leads to activation of transcription. This you should know, and you should know the name of the pathway. It is JAK-STAT pathway. The third type of receptors which are present on the plasma membrane are called as ion channels. Ion channels means these are the receptors, for example, present on the membrane, and these are the ions for which this receptor is specific. This much is clear. So these are the receptors, these are the ions which the receptor has a duty to take them inside the cell. But the receptor is closed. It will open only and only when a signal will come and binds with this receptor. So second, you can see this is a receptor. When a signal binds, then only the channel opens and allow the ions to move across. So these are called as ion channels. I can give one more name to this. These are also called as, many of you would be knowing, it is also called as ligand gated ion channels, isn't it? Because their opening is controlled by a signal, by a ligand. So we can also say they are also called as ligand gated ion channels. Okay, the very simple example for this is IP3 gated ion channels. So you can remember, for example, if this signal is IP3, It is present on, this type of receptor is present on the endoplasmic membrane, endoplasmic reticulum. So when this IP3 naming signal comes and bind, what happens? The receptor gets open and allow which type of ions? Calcium ions to move inside the cytoplasm. This is one example which you can remember. The most important receptors out of 4 are called as GPCRs in the short form, and the full form is G protein coupled receptors, or it can also be called as G protein linked receptors. It is one and the same thing. So let us see about this. When I say about GPCRs, the G protein coupled receptor basically involves 3 things. One is receptor, one is G protein, and one is effector. So these are the 3 main parts of this GPCR: receptor, G protein, and effector. And why it is called as G protein coupled receptor? Because this receptor working will be completed when G protein gets activated, and when it gets coupled with R, then only the G gets activated and then it activates the effector. So why it is called as G protein coupled receptor? So here you can see that when a signal binds to this receptor, it activates G protein. So they are coupled together, and after the G protein get activated, it activates the third member, that is effector. Effector means any type of enzyme generally. So because the receptor is coupled with the G protein for its activity, this is called as G protein coupled receptor. One question which comes generally, that the GPCR is dash transmembrane protein, fill in the blank. So G protein coupled receptors are 7-pass transmembrane protein. ये जो receptor है, this is 7-pass transmembrane protein. It means 7 times it crosses the membrane. And some points about G protein. When I say about G protein, first, why it is called as G protein? Because it binds with GTP or GDP. So guanine nucleotide, that is why it is called as G protein. Second point, the G protein is basically trimeric in nature. It is made up of 3 subunits: alpha, beta, and gamma. This alpha subunit has GDP or GTP binding domain. When these 3 units are together, that is alpha, beta, and gamma, GDP is joined with it and they are inactive. But when the beta gamma get dissociated and a GTP get binds, the alpha subunits become active. And one more point about alpha subunit is that it carries one more activity which is called as GTPase activity. It means it has the property of hydrolysis of GTP. It can hydrolyze GTP, guanosine triphosphate. Now let us see what is the mechanism, major points. So when you have a receptor, you have a signal, you have G protein, you have effector. When signal binds, the receptor undergoes conformational changes, and when it undergoes conformational changes, there is a site created for the G protein. So the G protein will come and bind. Here till now the G protein is GDP bounded. So when the G protein binds with this receptor, till that it was inactive, but the time it binds to the receptor, it then undergoes conformational changes such that the link between the alpha and beta gamma get detaches. So now what is the next step? Beta gamma get detaches from the alpha and the GTP is bind. So now the G alpha with the GTP is in active form. So what would be the next step? This G alpha GTP will go and activate the particular effector. When I say effector, I am talking about some type of enzyme. So G alpha will activate the effector, or you can say enzyme, When I say enzyme, what can be the enzyme? There are many types of enzyme. Let us take one example. If this G alpha binds with the effector, for example, enzyme is adenylyl cyclase. So what is the function of adenylyl cyclase? It will pick the ATP from the cytoplasm and it will remove the pyrophosphate, that is 2P, and it will convert it into cyclic AMP. So here you can see the reaction. Adenylyl cyclase, it will, if this is ATP, adenosine triphosphate, it will remove the 2P, that is pyrophosphate. You get a cyclic, the complete circle, adenosine monophosphate. So this is the, till now, this is the first product which is formed inside the cell. Okay. And after some time, when the GTPase activity get activated, GTP will be cleaved, again GTP will be formed, GDP, and they will again come to the first situation. Now, so this was the mechanism how the G protein coupled receptors work. The major protein in the whole GPCR system was G protein. In G protein also G alpha, which has actually the catalytic activity. So when I say about G alpha are of many types. Few let us discuss. One is GS alpha. S stands for stimulatory, which we discussed right now. And this GS alpha will activate a particular type of enzyme. Which enzyme? For example, adenylyl cyclase. So what would be produced? Cyclic AMP will be produced and its level increases. The third column heading is secondary messengers, or you can say second messengers. What is it? So that I would be discussing in the forthcoming slides. Second is G-olf alpha, that is G-olfaction alpha. So it means I am talking about smell. Okay. Here the effector enzyme is again adenylyl cyclase, and what is the messenger which is produced? Cyclic AMP again. The next type of G alpha protein is GT alpha, where T stands for transducin. The enzyme which is playing role here is cyclic GMP phosphodiesterase. So as the name suggests, it would be breaking the cyclic GMP. So what will happen? The cyclic GMP level will decrease. One more type of G alpha is GQ alpha, which basically act on the enzyme called as phospholipase C, and on its activity it will lead to the production of two molecules. One is IP3 and this is the second one, this is DAG. IP3 is inositol 1,4,5 triphosphate and DAG is diacyl glycerol. Okay, so these are few G alpha type of proteins. Now, so till now the story says a signal binds to the receptor, GPCR receptor, and GPCR activates protein, G activates a particular enzyme. If it is adenylyl cyclase, it will form cyclic AMP. Then what is the role of cyclic AMP? Now cyclic AMP in the cytoplasm activates one enzyme which is called as protein kinase A. So this is the structure of protein kinase A, which is a tetramer subunit where this dimer has a catalytic role and these two has a regulatory role. And the cyclic AMP when produced binds to this regulatory subunit of protein kinase A. So what will happen when the cyclic AMP will bind to the regulatory subunit of protein kinase A? The time it binds here, the bond between these two get removed and the catalytic subunit is free for its activity. Clear? So the next step is cyclic AMP binding and the catalytic activity is into action. Now, when I say catalytic activity is into action, it means what is the function of protein kinase A? Like all kinases, it will also phosphorylate when I use the word kinase. So the target protein, now the protein kinase is A, when it gets activated, it will act on the target protein and it will phosphorylate. There will be addition of phosphate group on the target protein. That is the function of protein kinase A. Now, kinases do add phosphate group anywhere. So, no. Protein kinases basically add the phosphate group in the protein at a particular target amino acid. Few examples are few proteins called as protein ST kinases. S stands for serine, T stands for threonine. Example is protein kinase is A, protein kinase C. C would be coming now. A we have dealt. So protein kinase A and C basically put phosphate either to serine or to threonine. Few kinases called as protein tyrosine kinase. It will basically phosphorylate the tyrosine amino acid. Few kinases have dual specificity. They do phosphorylation in any of the amino acid given 3. And there are very rare type of histidine kinases which basically do the phosphorylation on the histidine amino acid of a protein. Now, one example for this whole story we dealt till now, when, for example, there is a signal called as glucagon, which is a hormone. So when this glucagon hormone binds to the G protein coupled receptor, for example, in a cell, on the cell, if it binds to the GPCR, glucagon hormone, it will leads to activation of G protein. G protein will activate enzyme adenylyl cyclase and it will activate protein kinase A. This protein kinase A will do what? Now, whenever glucagon would be binding, the cell responses increase in the glucose level. So how it will be happening? So let's see. The protein kinase A, when activated, it will now, because it is kinase, it will do phosphorylation. So it do the phosphorylation of glycogen phosphorylase kinase. GPK is glycogen phosphorylase kinase. So, okay, the first step is done. GPK undergo phosphorylated, and after phosphorylation, GPK becomes active. So now this GPK will do the phosphorylation of GP. GP stands for glycogen phosphorylase. Okay, now GP is also phosphorylated, and glycogen phosphorylase on phosphorylation becomes active. So now what is the function of glycogen phosphorylase? It will cleave the glycogen, which is a polymer of glucose, you are knowing, into the monomer glucose units. So there would be increase in the glucose level. Simultaneously, if we see, there is one more enzyme which is called as glycogen synthase. And this glycogen synthase, when undergoes phosphorylation by the protein kinases, it undergoes inactivation. So this is a good story. When glycogen synthase will be phosphorylated, it becomes inactive. So there will be no synthesis of glucose. And here, by this process, the glucose level will go on increasing. So this is one example. Now, let us deal about second messengers, or you can say secondary messengers. What do you mean by this? So, the messengers which bring the message to the cell are called as primary messengers, and primary messenger, the response which is given by the cell first in the form of first product, it is called as secondary messengers. Here you can see the small molecules synthesized in the cells in response to the external signal are called as secondary messengers, which again plays role in the intracellular signal transduction. Basic are 5 types of secondary messengers: calcium, DAG, IP3, cyclic AMP, and cyclic GMP. Let us discuss the major points of these all. First, we will deal about DAG and IP3. If I say about DAG and IP3, the first point is, if this is plasma membrane, this whole is plasma membrane. IP3 and DAG are the two secondary messengers which are basically synthesized from a phospholipid called as phosphatidylinositol. So that can be the question, which secondary messenger is synthesized from a phospholipid? So they are IP3 and DAG are the two secondary messengers which are synthesized from the plasma membrane present phospholipid, which is phosphatidylinositol. Now what happens in plasma membrane? The phosphatidylinositol is present, and with the help of phosphatidylinositol kinases, they go on adding phosphate to this PI. So phosphatidylinositol, when undergoes 2-time phosphorylation, it becomes PIP2, that is phosphatidylinositol 4,5-bis biphosphate, PIP2. This is a normal process, but when the cell requires the two secondary messengers, that is DAG and IP3, the signal binds to the G protein receptors. Here the G protein is GQ, and it leads to activation of an enzyme which is PLC, and the PLC is phospholipase C. After the phospholipase C gets activated, it basically breaks down this phosphatidylinositol 4,5 biphosphate into from here. So you will get two parts, one IP3 which will move into the cytoplasm, and one is DAG 1,2-diacylglycerol, which is because this R chain, it will remain in the membrane. So this is the way how the two secondary messengers are synthesized from a phospholipid. Now, what is the role of these two? So basically, the role of IP3 here is calcium mobilization. Again, if we see PIP2 phospholipase C, it will break. There will be two formations, DAG and IP3. I said IP3 has role in calcium mobilization. Means what? This IP3 then goes to the cytoplasm, to the ER membrane, endoplasmic membrane, and the ER membrane has one type of channel which is called as IP3 gated calcium channel. So what will happen? IP3 is a signal. When the signal will bind, then only this calcium channel get opens, and the calcium from the lumen of endoplasmic reticulum moves into the cytoplasm. Here the IP3 role is done. Now the calcium present increasing in the cytoplasm binds with protein kinase C, which moves to the DAG. The mobility of this is increased by the calcium. It moves to the DAG and it becomes active, and then because it is kinases, it will do the phosphorylation of the target protein. So it will do the phosphorylation of the target protein inside the cell. This is one step. Again, there will be number of steps, and at the end the cell will shows responses in the form of cell growth or differentiation or transcription of or changes in the metabolism. So what is the role of DAG and IP3? They basically plays role in calcium mobility and PKC, protein kinase C activation. Okay. One more type of, we dealt about two types of secondary messengers. One is DAG, one is IP3. Third one is calcium. Calcium also plays role in signal transduction, as when cytosolic level of calcium increases, as we saw in the last slide also, it binds with specific type of proteins which are called as calmodulin proteins. So the function of calmodulin protein is to bind to the extra calcium present in the cytoplasm. Few points about calmodulins are they are dumbbell shaped proteins which binds 4 calcium at a time. So when this calcium binds to these calmodulin proteins, these complex of these two will activate calcium calmodulin dependent kinases, which are also called as CAM kinases. And these CAM kinases, when gets activated, because it is kinase, it will phosphorylate its target, and again the phosphorylated protein will do the further reactions, play role in the further reactions, and the cell shows responses in the form of cell motility, exocytosis, synthesis of neurotransmitters. So this is the figure which shows this is a calmodulin. These are the calcium binds. When these balls calcium binds with this calmodulin, they becomes calcium calmodulin complex, and this complex activates CAM kinases, and which CAM kinases on activation do the phosphorylation of the target protein. One more type of secondary messenger, which is one more type of molecule which plays very important role in signal transduction is nitric oxide, which is playing role in the synthesis of a secondary messenger, that is cyclic GMP. CAMP we dealt about DAG and IP3, then calcium, now it's cyclic GMP. So what is the role of nitric oxide? Now, whenever the cell needs to undergo relaxation of muscle cells or vasodilation, specific neurotransmitters, for example, acetylcholine is synthesized. Its signal binds to the receptor in the endothelial cell membrane. These GPCRs then will activate the effector. Here the effector is phospholipase C, that is PLC. Phospholipase C present on the plasma membrane, it will lead to the synthesis of IP3 inositol 1,4,5-triphosphate. This IP3 when activated, it will activate the calcium increase in the cytoplasm, and the increase in calcium leads to the binding of calcium to the calmodulin protein. This calcium calmodulin complex will activate the CAM dependent kinases. The CAM dependent kinases will leads to phosphorylation of the target protein. For example, if it is NO, nitric oxide synthase, this will become active and it will convert arginine into citrulline and nitric oxide. This from the endothelial cells moves to the smooth muscle cells, where they basically binds to the receptor, and their function is conversion of GTP. Whenever they binds to the particular receptor, the activation leads to conversion of GTP into cyclic AGMP, that is cyclic guanosine monophosphate. And this cyclic GMP then activates protein kinase G. And the protein kinase G which is formed is responsible for relaxation of muscle cells. So here cyclic GMP activates protein kinase G, cyclic AMP activates protein kinase A, which we dealt, and DAG and IP3 activates protein kinase C. All are different 3 types of protein kinases which we dealt. So you should remember. Now, GPCR, we dealt about secondary messengers, we dealt about GPCR. Now, GPCR's 2 major functions which you should know. One is light activates GPCR. Matlab in your vision also GPCR plays very important role. So what is the role of GPCR in the vision? So our eyes has rod cells in the retina, and these rod cells have GPCR complex which is called as rhodopsin, which has 2 parts, cis-retinal, which is obtained from vitamin A, and the opsin part, which is 7-pass transmembrane protein. These 2 combines to make rhodopsin. So whenever light enters this rhodopsin, which is acting as a receptor, the cis-retinal undergoes change and converts into trans-retinal. And when it converts into trans-retinal, it gets detaches from this opsin. And now we are left with the opsin, to which this receptor now binds with the G protein. So, and one more thing, that where are these rod cells present? So they are present in the outer segment, and these discs which you are seeing, these discs are basically containing the rhodopsin, that is the GPCR. Now, so when the light enters, the cis converts into trans, the opsin is free now, and the opsin will basically bind to the G protein. We are talking about GPCR, so obviously G protein will be there. Now, when I say about G protein, the G protein here has alpha, beta, gamma, and the alpha subunit is called as G alpha T. So T stands for transducin unit. The G alpha T, when undergoes activation, it will activate one enzyme which is called as PDE. You can write it is phosphodiesterase. So when the phosphodiesterase gets activated, what is the function? It will cleave the cyclic GMP. It will cleave the phosphodiester bond of cyclic GMP. So the cyclic GMP will convert into GMP. The cycle is broken down. And when cyclic GMP is converted into GMP, what will happen? The cyclic GMP level will be decreased. And cyclic GMP level will decreases, the channels which are ligand gated. Here CGMP is acting as a ligand which is controlling the opening of sodium and calcium channels. So when cyclic GMP level will go down, the channels which are opened due to this will get closed. So cyclic GMP gated sodium calcium channels will get closed, which will lead to hyperpolarization because changes in the charge in the cell. So then due to hyperpolarization, the message will goes to the brain and the response will occur. So here one important point is that in the vision, when the light is received, one type of secondary messenger is playing role, which is cyclic GMP, and the other point is its level is decreases. You should also remember the name of the effector, that is phosphodiesterase. Two major points. Similarly, when we talk about smell, that is olfaction, we can smell large varieties of different types of odors. We have the capability. It means your nose should have something which can differentiate between so many types of odors. So if I say we have olfactory neurons present in the nose, and they have cilias, and these cilias have receptors which are called as G protein coupled receptors. So whenever any smell comes, it is basically acting as a signal for the GPCR, which are called as olfactory receptors present on the cilia membrane. So whenever the signal comes, it will activate the G protein. The G protein here is called as G-olf alpha. The G-olf alpha, when activates, it will basically activate adenylyl cyclase. This adenylyl cyclase will activate cyclic AMP, not activate, it will leads to synthesis of cyclic AMP from ATP. When cyclic AMP level increases, it will open a cyclic AMP gated cation. Basically, it is sodium ion channels. So the cyclic AMP gated sodium channels will get open. It will lead to depolarization. The message through nerve impulse will move to the brain, and you would be able to differentiate the odor. Here, one more thing, the neurons which are present, they are large in number, and to recognize a particular type of odor, one neuron present below the cilia can recognize a single type of odor and more than single type of odor. Neurons also work in combination to differentiate the odor. So that is one more point you should know. This is all about signal transduction. Now let us discuss few questions which have appeared in your exams. These are the references which we have used. Now let us see the questions which are mainly important. The question number 1 says, which of the following signal molecule is not used for extracellular signaling? So again, question 1. Now you can write the answers by yourself for question 1 and 2. Then I will be telling the answer, so you can be able to judge yourself. So which of the following signal molecule is not used for extracellular signaling? So the answer would be cyclic AMP. Arrange the following sequence of extracellular signaling in the correct order. Now there are 4 options. You have to tell the order. Transport of signal to the target occurs first. Start of signal transduction pathway. Signaling cell synthesize and release the signaling molecule. Binding of the signal to the receptor. You have to arrange the steps 1, 2, 3, 4, the order. So what would be the order? The order would be 3, 1, 4, 2. Obviously, first the signaling cell will synthesize the signal. Okay. Then the signal will move to the target. Then the signal will bind to the receptor, and then the signal transduction will start. Let us see the third question. Mark the signal molecule which does not interact with the cell surface receptor. Signal molecule does not bind to the cell surface receptor. This means what? This means they are asking, name the signal molecule which binds to the intracellular receptor. So when I say this is a very good hint. Insulin, because it is a protein nature, it will be intracellular. No, it will be on the cell surface, cell membrane. Glucagon, same. Gastrin also peptide, it will also bind to the cell surface membrane. So who will bind to the intracellular receptor? So the answer is testosterone, isn't it? Because it is lipid in nature. Which of the following G protein takes part in the regulation of vision? Abhi, just now we have, discussed about the point. Which of the following G protein takes part in the regulation of vision? So vision is G stimulatory, G transducin, GQ, or G olfaction. Vision, which G protein? I think you would be knowing the answer. It is GT. T stands for transducin. Which of the following is true regarding a transmembrane receptor? When we talk about receptor, which point is correct? You have to say. It has both hydrophobic and hydrophilic regions, and the ligand binds on the hydrophobic regions. It has both hydrophobic and hydrophilic part, and the signal binds to the hydrophilic part. It has hydrophilic part only, or it has hydrophobic. So the correct answer would be, what do people think? I think you all would have given the correct answer. It would be this, the second wala, that it has both hydrophobic and hydrophilic regions, and the signal or the ligand will bind to the hydrophilic part. The next question, which of the following statements about G protein is wrong? Okay, false. So 3 would be correct, 1 would be false. Let us see. G proteins are involved in signal cascades. Yes, cascade is a chain of reaction which occurs one after the other. They bind to and are regulated by guanine nucleotide. Yes, that is why they are called a G protein. Second is correct. They become activated when bound to GDP. They must be active before the cell can make needed cyclic AMP. So the correct option is C. They don't get activated when bound to GDP. They get activated when they bound to GTP, guanosine triphosphate, isn't it? The next question. Cyclic AMP and cyclic GMP are derived from. So cyclic AMP and cyclic GMP, they are derived from what? So there are 4 options. ATP and GTP, so they are derived by the action of adenylyl cyclase and guanylyl cyclase. GTP and ATP by the action of adenylate and guanylate. ATP and GTP by the action of guanylate cyclase and adenylate respectively. And the fourth is none of the above. So the correct answer is ATP is responsible for the cyclic AMP synthesis, and GTP leads to the production of cyclic GMP. The enzymes are these two. One more question. The enzyme that catalyzes the splitting of phosphatidyl 4,5-biphosphate into 2 molecules of IP3 and DAG in the cell signaling is, you have to just remember the name of the enzyme. Which enzyme breaks this PIP2? So I think the options are quite easy. Phosphokinase C, phospholipase C, phosphodiesterase C, or lipokinase. So I think, and you would be thinking the same, I hope so, the answer is B. So it will be phospholipase C. The second last question, which of the following is the secondary messenger? Very easy. Inositol 1,4,5 triphosphate, which we say IP3. Second says DAG, that is diacylglycerol. Third is phospholipase C, which is short as PLC. And the fourth says both A and B. So the correct answer would be D. IP3 and DAG, these two are secondary messenger. The C wala is enzyme. The last question, what happens in the smell? Or I can say, what signal transduction process basically occurs when I talk about smelling process? So there are four options. Whenever you smell something, the cyclic AMP level increases, it decreases, cyclic GMP plays the role and decreases, or cyclic GMP increases. Answer is very easy. Answer is 1, that is cyclic AMP level increases. So this is all about the topic. If you have any query, you can write in the chat box, or maybe the next speaker if has come.
Shweta ma'am, I can give them rights for 2 minutes if they want. I'm allowing them to unmute themselves and ask questions.
Okay, ma'am, ask them to write in a chat box. Maybe unmuting, they all will speak then.
Okay.
Or they cannot write na in chat box.
There is no chat box, I think.
Okay.
I've given them rights so they can unmute themselves and tell their name and ask questions if they want.
Okay then. Okay. I think everything is too much clear to them. So, okay, ma'am, then I think it is clear.
Okay.
Shall I leave now or?
Yes, ma'am.
Okay, thank you.
Okay. Thank
Thank
you
you,
so much, ma'am.
everyone. Yeah, thank you, everyone, and all the best for your examinations.
Students, your next lecture will commence after half an hour. You have a break. So we'll all gather together back at 4:25 p.m., right? It's 5 minutes. It's 3:56 by my watch. So please come back at 4:25 so that we can assemble and the next speaker can start, right? You can leave the meeting and please come back after half an hour. Thank you so much for joining with us. We'll meet again for the rest 2 lectures for the day. Thank you. Hi, welcome back to the second session of Gsbtm sponsored crash workshop on national level competitive examination for final year UG students. I'm sorry for the delay. There was a little trouble with our audio sound. So the next speaker for the day is Dr. Mehul Dave. Sir is Associate Professor at Indian Institute of Teacher Education, Gandhinagar. Sir has a teaching experience of 19 years and has qualified almost all the national level competitive exams like UGC NET, CSIR NET, GSET, etc. Sir will be talking about evolution, and Sir has this immense talent of explaining the most difficult topics in the most easiest manner. I hope you enjoy learning with Sir. I request Sir to kindly start with his lecture. Thank you so much.
Thank you very much, Ragini madam. And just confirm whether my sound is audible to all of you or not.
Yes, sir, you're audible.
All right. Hello,
Okay.
everyone. The topic which we will be discussing for an hour, that will be something like protobiogenesis. Anyone knows what is the meaning of that word, protobiogenesis? You can unmute yourself and you can give answer if you know. What is protobiogenesis? If not, then I'll quickly, I will not waste a moment. Proto means primitive, bio means life, and genesis means origin. So protobiogenesis means origin of life on earth, right? So initially we want to talk about that origin of life, and then slowly we will move towards evolution and various theories of Darwin, Lamarck, Hugo de Vries, and then Dobzhansky, neo-Darwinism, and all right. So for that we shall look to certain so many interesting things, content, everything. I'll just share one presentation in a moment. Is this visible? PowerPoint is visible, madam?
Yes, sir, it's visible.
All right. And students are able to unmute themselves and interact or not?
No, sir. We have muted them all, and we can have the session, question answer session at the last if you want.
Fine.
Okay.
Fine. All right. Okay. Just I'll keep
Thank
that in mind so that I'll not keep on asking anything to students. All right.
you. Okay,
So,
thank you.
as you know, father of evolution, everyone knows, Charles Darwin, right? But few of you might be knowing father of microbial evolution. Remember that name also, father of microbial evolution is the one who explained three domain classification of all living organisms. Now, probably the name is in your mind. It is Carl Wood. So, father of evolution is Charles Darwin, and father of microbial evolution is Carl Wood. In our discussion, we will discuss everything about evolution of higher forms of living organisms, and lower organisms also. Okay, so we will discuss about origin of life, evolution, Lamarckism, Darwinism, and everything. Fine. Madam has given my introduction. Now this is father of evolution, as everyone knows. So we wish to begin by mentioning his name, looking to his gorgeous photograph, Charles Darwin. Fine. And the one who provided neo-Darwinism, modern theory of evolution, was Dobzhansky, and he told nothing in biology makes sense except in the light of evolution. So whatever branch of life science we study, right, it connects to evolution in one or other way, that you have to understand. So everything is understood in the light of evolution, right? So there were so many myths on the earth, okay, before Darwin's era, okay, there were so many myths on the earth. For example, species were not linked into single family tree. Everyone understood that they are individual species. And in Christianity in Europe, people believed that earth is only 6,000 years old. Today we know that earth is actually 4.6 billion year old. That is 46,000 lakh year old the earth is, right? But the belief was wrong, the myth was wrong. And it was also understood in the era 200 years ago that the world is stable and it is unchanging, whereas we know that it is very much dynamic. Life is dynamic, it's changing every day. Even our skin is changing every day, just like a river. Living organisms, their properties, their character, the antigenic profile of coronavirus, we know it is changing day by day. All right, so there is evolution everywhere in the earth when we talk about living organism. Okay, so Evolution means cumulative changes in the heritable characteristics of a population observed over time. So, if we learn something, if we increase our knowledge, if we gain degrees, it is not evolution in biological sense, because it should be heritable characteristic to be changed. All right. So when something in our DNA is modified, something which we transmit to our next generation, then we have to call it as evolution, and it is not related to getting better. Mind well, evolution is actually dependent 100% on the environmental changes. So if environment goes towards X pathway, then evolution will occur in X way. If environmental changes occurs in the Y manner, then evolution will occur Y way. All right. So, and DNA changes whatever occur during our lifetime, they are passed to our descendants. So evolution is when organisms change over time. So modern organisms are actually descended from the ancient ones. So you can see this is a very beautiful picture that explains us something about evolution. All right. Evolution is everywhere. For example, aquatic organisms told amphibia, and amphibia told the other one, right? That reptiles, for example, don't walk away when I'm talking to you, but evolution took place and life migrated from the ocean to the land, probably, and all living organisms, they have one formula: eat, survive, and reproduce. But human being, because we think that what is it all about, therefore we are, in little bit faster in the evolution, particularly whenever intelligence is connected. Therefore, our scientific name is Homo sapiens sapiens, right? Sapiens, the word relates to intelligence. Okay, so evolution is in the mobile also, evolution is in the technology also, evolution is in the warfare also, evolution is everywhere in the life. And the cartoon says that it seems that human race is evolving because the baby is born with bulletproof skin. So there is evolution, and evolution, mind well, it's not hypothesis, it's a theory. The word natural selection is a theory, just like theory of gravity, it is well proven theory. But mind well, the aim of life told by various biologists is, the aim of life suggested by biologists is continuity of life. Our aim on the earth is only to continue the life on the earth. Mind well. All right. So quickly we'll talk about protobiogenesis, origin of life, and all the cultures have creation myths, right? Somebody, in some particular Egyptian culture, it was sun god who creates everything. In China, Pangu. In May culture, May Sanskruti, right? That was, serpent god who creates life. In Zulu, in Judo, Christian, in all cultures, there are different, gods or divine forces according to them. They have created life on earth. Even in Hinduism also, there is a concept of Brahma, Vishnu, and Mahesh. Brahma is the creator, Vishnu is the manager, and Mahesh is the destroyer. Now, that is the concept in Hinduism also, right? The father of biology, as we know, Aristotle. Remember, father can also be sometimes wrong. Aristotle, no doubt, is father of biology, not any branch of biology, but all over, father of biology is Aristotle. He lived in the era around, 2.5 K ago, around 2400 years ago, he lived on the earth. He gave so many basic principles. He told, about oviparous and viviparous. He suggested about various systems of the animals. But Aristotle was wrong in one way. Aristotle believed in abiogenesis. Aristotle believed that life can be created from non-living things. Now, that is the point where Aristotle was wrong, but we will, will consider, even though contribution of Aristotle as a mammoth contribution, because whatever he did when there was no science, when there was no knowledge, 2500 years ago. Okay. So that is a quick note about Aristotle. Now, there were, mysteries like cosmogenesis, right? Somebody suggested life may have come from other planets. Somebody suggested biogenesis. Somebody believed in evolution. Somebody believed in various theories. These are certain misconceptions and myths about the life. As you know, survival of the fittest. Now, many times we attribute this particular phrase to Charles Darwin, but it was not suggested by Darwin. It was suggested by a philosopher, Herbert Spencer. Mind well. All right. So that is also one correction which you should do. Various theories regarding origin of life were like this: abiogenesis, life is created from non-living things. It is also known as spontaneous generation theory. There is another theory called biogenesis. It was proposed by Francisco Redi, and according to him, the life can be created from only living organisms, and Cluse and Schwann supported, and Louis Pasteur by developing a Schwann neck or goose neck flask, swan neck flask. Louis Pasteur proved that life can be created only from living organism. Recently, there is a correction in that. Recently, in 2015, Craig Venter at Celera Genomics, he created artificial living organism from, synthetic genome, and the name of that organism is probably some of you might be knowing, right? Mycoplasma laboratorium. So again, abiogenesis comes in the picture, mind well. But I'll move ahead. Various theories included like meteorite theory. So life has come from meteorite shower. There is theory of eternity. Theory of eternity means there was life on earth, there is life, and there will be life. Then there was another theory, catastrophe theory. In Hinduism, we call it Satyug, Dwapar Treta Yuga, and Kali Yuga. So that are frequent catastrophe, and there is restarting of everything. That is theory of catastrophism. Theory of special creation. It means divine forces, God, they have created life on earth. And then theory of organic evolution. Probably all of you know that it was proposed by Halden. All right, JBS Halden. Okay. So that is organic evolution theory. This is, Opperin-Miller hypothesis, and later on, Urey-Miller's experiment. Everyone studying biology and reached to this graduate level, you have studied this experiment. So Urey-Miller proposed and proved in 1953, the same year when the double helical structure of DNA was proposed. In that year, it was proved that methane, ammonia, water vapor, hydrogen gases, and the energy from lightning that resulted in the development or creation of amino acid, and this amino acid later on might have joined, might have developed some primitive proteins, they acted like enzymes, and catalytic reactions might have been started, and by that way metabolism started and life could begin. So life began from chemicals. That was Urey-Miller's theory or Urey-Miller's experimental proof. Now this is very interesting. If you see that life on the earth as it is today, it is actually the result of 4.6 billion year process, 46,000 year process. If we shrink that entire process in one day, in 24 hours, then the picture will be something like this. And here in this diagram, 1 second is equal to 52,000 years, and 1 minute equal to several lakh, and 1 hour is actually equal to several million years. And life started 12 o'clock yesterday, and right now it is, bell is ringing of 12 o'clock today. Then you understand that at around 3 a.m. late night, there was formation of rocks. Prokaryotic organism came into existence at around 5 to 6 a.m. Oxygen entered at around 1 to 2 p.m. Eukaryotic organisms, they might have developed around at 4 to 5 p.m. Multicellular fossils around 8 to 9, only few years ago, few hours ago. Land plants, dinosaur came into existence and then become extinct between 10 to 11 p.m. and right now it is 12 o'clock. Then, just before 30 seconds, Homo sapiens, and just before 5 seconds, cultured Homo sapiens appeared on the earth. So we are very, very new, very, very junior, extremely, modern product of evolution, what we are today. And microorganisms, plants, they are quite senior to us. That you should remember. Evolution is actually a science. Evolution is studied on the basis of fossil records, by studying the adaptation, by studying geographical distribution, by comparing DNA sequences. We study evolution, and evolution is the only theory. Mind well. I'll go ahead. Evolution, original 17th century definition was from the word evolutio, that is unrolling, dhime dhime khultu jai, right? That is evolution. Okay. And earlier it was believed, that sperm or egg might be containing some structures like a human body, and so that all myths were there when there was lack of knowledge. Okay. So evolution, actually today what should understand is, evolution is a transformation of population between the generations. Okay. There is an important word, transformation. Okay. So various scientists that should be known, that should be considered here: Aristotle, then Buffon, for example. Then two important contributors were Hutton and Lyell. Cuvier also. Hutton suggested that changes in nature are gradual. Lyell suggested that all the changes are gradual, and he suggested a lot of fossils. Cuvier suggested that species are recreated after catastrophe. And then two very big names in the theory of evolution are Baptiste de Lamarck and Charles Darwin. Lamarckism and Darwinism, we will discuss them in bit more detail, but we should not forget the contribution of Carl Linnaeus also. Okay, so someone has told that God created and Linnaeus organized because he's father of, as you know, father of taxonomy, Carl Linnaeus, and there's a lot of contribution of Carl Linnaeus in making us aware about evolution because he organized the living organism, classified them so that we could understand the transformation occurring in them. Okay, the book which he wrote was Systema Nature or Nature. Remember that also maybe sometimes asked in some question. And then Charles Darwin, quickly I'll tell something about him. He was actually doctor by a degree, but he didn't like performing medical practice, so he didn't work. He was moving here and there, actually we can say. And suddenly he got an opportunity to go on a world tour in a ship. The name of the ship was HMS Beagle, and the ship brought him to Galapagos Island. Okay, so name of the ship can also sometimes be asked in some competitive exam. Remember HMS, that is Her Majesty's Service. Her Majesty's Service, right? HMS Beagle. Okay, so Darwin went on the islands known as Galapagos, and he raised so many questions. Actually, he went as an attendant on the ship, but the captain gave him extra job of environmental officer, seeing his degree. And Charles Darwin went on various islands and geographical locations. He collected certain evidences, thought about them, came back, and after a few years of research, he could publish a book known as On the Origin of Species. And there, comes the entire story of evolution. Okay, so this is the book in 1859. It was published by Charles Darwin. And these are certain other points about Darwinism. We will discuss them in bit more detail. Europeans believed that world is only few thousand years old, but Darwin opposed that, and European people didn't like it, suggested him to go away from Europe, and that entire is a lot of story. Darwin thought all these things you can read on the screen. So quickly, this is the photograph of the ship where he went. He went on the world tour when he was only just like you, 22 years old. The captain of the ship was Robert Fitzroy. Remember his name too. And this is the entire route of the ship travel. Okay, and remember the Galapagos Island in the left side, you can see in the western coast of South America, there lies the archipelago actually. It is Galapagos. So number of islands together, it is archipelago. But remember, my dear student, when Darwin was working in Galapagos, at the same time in Malay archipelago near Malaysia, there was another great scientist, Alfred Russel Wallace. He was also working with the similar concept. He also came with similar conclusion, and he also believed in natural selection theory. Okay, so both the scientists suggested natural selection, but Darwin could publish his finding earlier. Therefore, Darwin was, given more credit, but we must not forget the name of Alfred Russel Wallace. All right. Okay, Darwin's travel revealed that there is a lot of diversity, but there is some connection among all the living organisms. There is, so there is something like classification. There is some connection. There are certain groups which he could understand, he could find, and he suggested that there is natural selection. Nature selects, the one who is fittest to survive in the nature. In Galapagos Island, there were 4 main species. You can read the name, that can also be asked in competitive exam. 4 species were mainly studied by Darwin to propose the theory of evolution. One was land tortoise, or what we call them as the Abingdon tortoise. The second one was Finch's bird. Third studied was blue-footed booby, and fourth one was marine iguana. So these 4 animals, you can see the Abingdon tortoises and iguanas. You can see the photograph of blue-footed booby, and in the upper right side it is Finch's Then you can see the tortoises and iguanas also. So these animals were studied by Darwin to explain the theory of natural selection. Okay. In different islands, there were different shape of the shells of the tortoises, and Darwin could correlate them with the food habit and came with some conclusion. Remember, in the same way, probably you have studied in your school that Darwin finches, these are the birds having similar body, but there is variation in the shape of the beak, and the shape of the beak is dependent on the food available. So on each island, there is a new variety of the finch bird. So Darwin suggested a word known as adaptive radiation. All come from same point and then go diversifying in a different direction. These are the Abingdon tortoises which he studied. He came back, he thought a lot, and he proposed the hypothesis that separate species may have arose from original ancestor. Remember? Then in 1859, he could publish this book on the origin of species. There is a lot of interesting thing about this book. I would suggest all of you to read that book, okay, on the origin of species. In certain national level book fair, this book is sold in very low price. It is sometimes available in certain internet resources also. Remember Alfred Russel Wallace? He wrote to Darwin about the theory of evolution that was similar to his. So Russell and Darwin, they were contemporary scientists. They believed in the similar theories. Remember name. This is the title of the book. This is the title of the book, new edition. And various people who influenced Darwin was Linnaeus, Lyell and Hutton, Cuvier, Lamarck, Alfred Russel Wallace. Remember these names too. And Thomas Malthus suggested there is a population growth. If there is unlimited resources available, then population always tends to grow. And when population grows, there is struggle of existence. There is limitation in the resources. As soon as there is limitation in the resources, there will be struggle, there will be competition, and in the competition, the one who is fit will survive, the one who is unfit will die. But remember, here fitness doesn't mean muscular fitness. Here fitness means reproductive fitness. The one who has higher reproductive power will survive. The species who has lost reproductive power may not survive. Okay, so this is Malthus. So Malthus suggested in the population growth, remember, population and struggle. Remember his name. If some question comes to you, then correlate the population and struggle with Thomas Malthus. Okay. Hutton, forces change Earth's surface shape. This is the suggestion by Hutton. Changes are slow. And this is Lyell, who published the book Principles of Geography. And see, I'll show you a picture. Yes, this one. So you can see various strata of the Earth's surface, and the fossils collected from different strata, they come from different era of the Earth. Therefore, if we collect fossils from the Cambrian strata, from Devonian strata, from Mississippian strata and Pennsylvanian strata, and if we arrange them, we can definitely understand the order of evolution, right? In which direction evolution occurred, that could be understood. If some of you knows, that how can we study evolution in just a very limited time? Actually, the story of evolution is 4.6 billion year old. But how can we get a quick view of evolution? Where can we see evolution? This is my question at the end of our lecture. I wish some of you answer this, that where can we see a nutshell, a very small story of evolution of living organisms that happened on the earth in the course of 4.6 billion years. Where can we see it in a nutshell? And what is the name of that particular theory? If some of you knows, I would be very happy to understand that, right? So that is one question. I'll go ahead in the presentation, Lyell and Hutton, and then Lamarck, punchline, my dear students, remember punchline of Lamarck's work was, inheritance of acquired characters. Whatever we acquire in our life, we pass on to our generation. That was the suggestion of this particular scientist, Jean-Baptiste Lamarck. Is it true? Whatever we gain in our life, for example, if some person who is having white skin tone lives for years in African country, his skin is tanned, becomes blackish, and then the children of that particular person, are blackish or fair in color. That is the question. According to Lemach's theory, it should be blackish. Okay. But you know that it's not so. And whatever we acquire in our lifetime may not be passed to our next generation. So that is the failure of Lemach. And Lemach's theory was corrected by Darwin. So what is the punchline of Lemach? Remember, my dear students, I'll speak once again. The punchline of Lemach is inheritance of acquired characters and use and disuse of the organs. This was, among the theory of Lemach. Okay. So, main principles suggested by Lemach, first was wrong. He believed in abiogenesis. Second, that organisms have innate power to progress towards more complex and perfect form. No, not like that. Third proposal of Lemach was organisms have inner disposition to adapt their characteristic in response to changes in the environment. That is partially correct. Point number 3 is. Fourth one is the main inheritance of acquired trait, and that was Lemach's point, which was totally rejected by Darwin. Okay, so Darwin suggested if a muscular male marrying a muscular female, will their children be born with six-pack body structure? Whatever we gain in our life, do we pass on to our children? No. What we pass on is our DNA sequences, not our muscles. All right. So inheritance of acquired character is not always true. So Lemach suggested this kala nature word, it's a ladder. Okay. Okay. Very interesting. Elongation of giraffe neck was explained as per Lemach and as per Darwin's theory. But see in the picture, Darwin-Wallace theory. As per Lemach's theory, remember, slowly and slowly, generation to generation, the neck length of the giraffe increased. થોડી વધતી ગઈ, like that, it was Lemach's theory. કારણ કે થોડોક વધારો next generation માં ગયો, next માં ઓર વધારો થયો. That was Lemach's theory. But Darwin and Wallace suggested that it's not like that, but because of genetic polymorphism, there were giraffes with all sizes of neck, and the one who had longer neck survived. That is natural selection, the one who had shorter neck died. So there is a drastic difference in the Lemach and Darwin's theory, and probably now you understand that Darwin's theory is the right one. Okay. So Lemach's theory na main findings, just you can read it. I explained already. This is Lemach's theory, slow elongation. All right. And you can see the Darwin's theory, that the one who has shorter neck died, one who had longer neck survived. Lemach and Darwin's theory, just a comparison, just you can see and try to understand. All right, I'll go ahead. Sorry, this is Lemach's theory. He was a Frenchman and Darwin was an Englishman. Remember this point too. I'll go ahead in Lemach's theory. Use and disuse. The organs which are used will elongate. For example, there are so many vestigial organs in our body. Namely, the first one, the vestigial organ is wisdom tooth, as you know. All right. Then one of our eyelid is also vestigial. Last finger of the foot is also a vestigial organ. And the nipples on the male chest, they are also vestigial. There are several appendix, as you know, these are vestigial organs. They are not used, and they are slowly becoming smaller and smaller. So in one aspect, Lemach, this particular point is correct. All right. Evolution of acquired trait, it is wrong. So this theory is wrong. Now we'll quickly come to Darwin's theory, and that is based on studying the evidences of evolution. The evidences which are studied to understand natural selection are fossils, geographical distribution, homologous body structure, similarities in early development. There is evidence number one, biochemistry. For example, basic similarity in all living organisms. The genetic material is nucleic acid. Some viruses may have RNA, others have DNA. So nucleic acid is universal genetic material. It means all have come from same point. All follow glycolytic pathway. So all have come from same. All have enzymes. All glycolysis begins with hexokinase only. There is conversion of glucose into glucose 6-phosphate and fructose 6-phosphate and fructose 1,6 biphosphate, dihydroxyacetone phosphate and phosphoglyceraldehyde and so on. All living organisms on the earth, whether plants, animals, microbes, viruses also, they all follow glycolytic pathway. So it means they have come from common ancestors. Then there is similarity among the genes also. For example, there are minor differences which we sometimes call as SNP, single nucleotide polymorphism. So, there are a lot of similarities among genes. It means all have come from common ancestors. Comparative anatomy can also explain us that there is evolution. So this is skeleton of human and gorilla, but they have a lot of similarity. Only the sizes and the shapes may be different, but the basic organization of the skeleton of the species who are nearby in the classification tree or phylogenetic tree, they are similar. Okay. Then homologous organs. So we'll call this as divergent evolution. I'll come to that point. Vestigial structures. For example, there are several vestigial structures mentioned here. Coccyx, for example, appendix, say for example, wisdom teeth, and nupetal membrane, as you know. Okay, fossil records can also explain us about evolution, which has taken place. Lyell and Hutton, remember these two names? And transitional fossils, in other words, we can call them as connecting links. So Archaeopteryx is a connecting link between reptiles and birds. So by understanding that connecting link, we can come towards the understanding of the direction of evolution. For example, if you are traveling from Rajkot to Ahmedabad and if you stop at the Limdi, then it is a connecting link between Rajkot and Ahmedabad. So in Rajkot, Gathia and Sambaro. In Ahmedabad, Gathia and Kadi. But if you stop at Limdi, then you will get Sambaro and Kadi both. So Limdi is a connecting link. If you just wait at Limdi, you can understand that this is a connecting link between Rajkot and Ahmedabad because there are dual properties. All right. So connecting link is having dual property. There are a lot of connecting links. So that is transitional fossils. And then geographical distribution of organism can also guide us, can also suggest us that evolution has occurred. Antibiotic resistance, will call this as anthropogenic evolution, man-made evolution. All right. So evolution occurs because of human influence. For example, excessive use of antibiotic has resulted in the development of antibiotic resistant bacteria, like here mentioned is like staphylococci. In the same way, excessive use of DDT has resulted in the development of DDT resistant mosquitoes. So that is anthropogenic evolution, man-made evolution. Even domesticated sheep or domesticated cows or horses, they are also anthropogenic evolution. Then tube-rail mosquito, it's also an example of anthropogenic evolution. Right. So there are a lot of evidences of evolution. Fossils are one of them. The study of fossil is known as paleontology. My dear students, remember sometimes the name of the branch of biology is also asked in certain competitive exams. There are a lot of evidences which I have mentioned. This slide suggests them. We have so many things to discuss. Therefore, I am just going quickly ahead in the slides. Relative dating. So here name again comes Lyell and Hutton, two scientists. Carbon dating, Archaeopteryx, fossil formation. There are a lot of evidences, right? This is Archaeopteryx, which is a connecting link between reptile and bird. So there are, you can see in the diagram, bird-like features are also there. Reptile-like features are also there. And this is silkant, the fish, okay, which lived in the earth 40 million years ago, and it could live little bit without water also. So this fish may be a connecting link between fishes and amphibia. And then comes very interesting homologous body structure. We call it as divergent evolution. Okay. So see further. This is, forelimb of human, cat, whale and bat. There is humerus, there is radius, ulna, there is carpal, there are metacarpal and phalanges. But you can see we use this forelimb for work, cat uses for walking, whale uses for swimming, and bat uses the same for flying. All four are quite different, but there is basic similarity in the anatomy. There is basic similarity in the bone arrangement. It means they all have come from common ancestor, and today we know that they all four are mammals. They have come from common ancestor. The first mammal on the earth, if some of the student can tell me, I'll be very happy, but you are unable to unmute yourself, so I'll give the answer. The first mammal on the earth was a mole, was a shrew, જેને આપણે છછુંદર કહીએ છીએ. We all are descendants of that particular mammal. આપણે એમાંથી ઉતરેલા છીએ, right? તો આ homologous body structure છે, it is divergent evolution, diversification. So from common point, all have moved in different direction. For example, all of you are TYBSc students. Later on, for your post-graduation studies, student number 1, all of you are right now, Indian, all of you are Gujarati, for example, all of you, are, for example, Kathiawadi. And then if you settle in different countries for your PG study, then there will be variation in your character. Some lives in USA, some goes to Japan, some goes to Australia, some goes to Afghanistan, for example. So after 5-7 years, you all will be, or after 10 years, you all will be having some variation, some changes in your lifestyle, in your belief, in your food habits. But when there is Jana Gana Mana, all of you will stand up because internally you all are same. Basically, you all are same, right? It means there is divergent evolution, basically same, but functionally different. This is divergent evolution. And then there will be another word, convergent evolution. We'll come to that also, but you can see the diagram of divergent evolution, homologous body structure. Convergent evolution has homologous function. It means convergent evolution, it means all are basically different, but today functionally similar. See, again, divergent evolution, basically similar, functionally different. Convergent evolution, basically different, but functionally coming together. So, divergent evolution goes like this. Convergent evolution comes like this. Fine. So in convergent evolution, I'll give you an example. Say, for example, you imagine that there is a insect like butterfly, there is bat, and there is bird. All of them can fly. Butterfly can fly, bird can fly, bat can also fly. They all have wings. Their wings look maybe similar in their function, but basically all of them are different. Ne? Butterfly belongs to arthropoda, and birds are, aves. Bat is mammals. All are having functional similarity, but basically, structurally, anatomically, they all are different. It is convergent evolution. Okay, so these are diagrams of divergent evolution. Adaptive radiation, starting from the common point, going in different directions. So it is convergent or divergent? Divergent. For example, marsupial ancestors resulted in the marsupial, like kangaroo, like having abdominal pouch or brooding pouch, what we call them. So marsupial ancestor resulted in development of marsupial wolf, cat, an anteater, rat, kangaroo, wombat, bandicoot, koala, mole and sugar glider. All બધાની પાસે brooding pouch છે. એટલે બધા common ancestor માંથી આવે છે. Like that, we have to understand. All mammals on the earth and all marsupials in Australian continent, for example, and all mammals in the Asian continent, they both, suggest that they have come from common ancestors. This is divergent evolution. But if you compare them, it is convergent evolution. If you compare Asian mole with marsupial mole, then they are functionally similar but structurally different. So it is convergent. So left column is divergent evolution. Right column is also divergent evolution. But if you compare left and right column, then it is convergent evolution. Very interesting. Okay. Okay. We have seen vestigial structures, so I'll move ahead. This is, embryonic similarity, embryological evidences, comparative embryology, fishes, reptiles, birds, and humans. Their early embryos are common because they all are coming from common ancestors. You can see this early embryo and later on further development of shark, lizard, chicken, pig, and man, But early embryos are similar. Later on, the variations come. So all of these are evidences of evolution, and all of them point out that natural selection is a correct theory. This is Darwin's theory suggesting adaptive radiation in the finch bird. You can see this. I've explained this further, so I'll not repeat it. Seed eater, bird eater, insect eater, cactus eater, all finches, they have similar body but different shape of the beak. Actually, there are 14 species today developed. That is divergent or convergent evolution? It is divergent evolution, mind well. See this entire phylogram, Galapagos finches in the different islands, scientific name, shape of the beak, everything is mentioned in this chart. Okay. So this is Darwin's conclusion. Natural selection is the word, is the buzzword, is a very, very important word. You can see the red lines written. There are so many other examples of natural selection and artificial selection too. Okay. So the Darwin's theory went into this point: struggle for existence, survival of the fittest, and natural selection. All right. So you understand all these things? So overproduction of offspring. So population growth, there comes the name of scientist Malthus. Variation, and then selection. And the one which is selected slowly adapts. This is artificial selection. Nature provides variation, but we select. We select the one who are obedient. Therefore, it is domestication. Farmer breeds only his best livestock, and slowly we have developed the cattle who give very huge amount of milk production, or the cattle having high disease resistance. So this is artificial selection. This is anthropogenic evolution, man-made evolution. There is one very interesting example of London moth, which is known as industrial melanism. We will come to that point also. Survival of the fittest. Remember, fitness is not physical fitness, it is reproductive fitness, as I told you earlier. All right. So there are various types of adaptations: anatomical adaptation, maybe physiological adaptation, and sometimes behavioral adaptations also. So individuals having low level of fitness, they die, do not reproduce, and those with high level of fitness, they survive, they reproduce. Therefore, they pass on the character to the offspring, and by that way, there is survival of the fittest. Summaries of Darwin's theory is like that. You can quickly go through the entire summary. I'll give you a time of a moment. Just read it out and you will definitely understand. I have passed on this particular literature to you so that you can also read it at your ease, at your convenient time. Okay. So, summary. Organism differ, variation is inherited, right? Organism produce more offspring than survive. So there comes environmental resistance and biotic potential and carrying capacity. These words of environmental science to be remembered. Organism compete. Those who have advantages to survive, they will pass the advantages to their children. But mind well, these are only genetic changes that are passed on. Okay, morphological changes may not be. Behavioral changes may not be. Species which are alive today are descended with modifications from common ancestors. So descent with modification, right? There is always descent with modification. Okay, so there is long summary of Darwin's theory, but in a nutshell, you can see that there are red and there are green beetles, but the crow likes only green beetles. Therefore, red have survived. They have got an advantage, evolutionary advantage. Therefore, slowly and slowly, the number of red beetles will increase in the population, and ultimately, red population will be there, or brown beetle population will be there. So all the species have occurred which have what we see today on the earth, they are like that only. What are the four animals? These are four animals studied by Darwin. Again, I'm coming to that line: Abingdon tortoise, Darwin finches, blue-footed bobby, and marine iguana. Remember these four animals. So ancient armadillo and present day, there is a variation. Okay, there is a change over time. Darwin asked, why should extinct armadillos and modern armadillos be found on the same continent? Today also we see that, So always remember, we have descended from the apes. The apes are already there today. Is there a relationship between the environment and what an animal looks like? Answer is always yes, there is a relationship. Okay, so earlier ideas of Lamarck are wrong. And Darwin's view of evolution, giraffe that already have long necks survive better. No, that is true. There are words like variation, selection, survival, reproduction, inheritance of fit or fitter traits. So this theory is correct. So Lamarck's view of evolution is wrong. Darwin's view of evolution is right, but certain aspects of Lamarckian theory are correct. So Lamarck's theory is partially correct, right? Now we will come to another point: modes of action of evolution or results of evolution. There are three ways in which evolution can result: stabilizing, directional, and disrupting. There are three ways. For example, this chart suggests percentage of marks. For example, some students in the class have 90, some students in the class have 50, but most of the students, they score 70% marks. This is just an example, all right? Now, if the university changes its examination strictness and makes the exam very strict, makes the passing criteria very high, all students will have to work harder. Therefore, slowly and slowly, the percentage distribution graph will move towards right side, isn't it? 70 વાળા વધારે મહેનત કરશે એટલે એને 80 આવશે, 50 વાળાને 60. ગ્રાફ છે એ રાઈટ સાઈડ શિફ્ટ થશે. So this is directional selection. એનાથી ઊંધું પણ થઈ શકે. યુનિવર્સિટીના સ્ટાન્ડર્ડ્સ બહુ નબળા થઈ જાય તો ગ્રાફ મેં માઇગ્રેટ ટુવર્ડ્સ લેફ્ટ સાઈડ, એને પણ આપણે ડિરેક્શનલ સિલેક્શન જ કહેશું. ઓકે, સ્ટેબિલાઇઝિંગ કોને કહેશું? જસ્ટ ઇમેજિન, ફોર એક્ઝામ્પલ, આને માર્ક્સ તરીકે જ કન્સિડર કરો. કન્સિડર જસ્ટ લાઈક માર્ક્સ ડિસ્ટ્રીબ્યુશન. તો 70% વાળા students say, I'll go to Beck's previous slide only. 70% students, they have all advantages. 90% they're very studious, they don't enjoy life. 50% they're very poor students, they enjoy life but they don't go in the next standard because they fail. 70% students, actually they enjoy life also and they pass also. Therefore, they go in the next standard also and enjoy life also. They participate in sports and cultural activities also. Therefore, 90% and 50% all are tempted to come towards 70% marks. But then 70% jeva thawun hoy, then the graph will become narrower and the shape of the graph will be like this now. It is stabilizing selection. I think you understand from this metaphor. This is the graph may migrate towards any side, graph of distribution of height or anything, percentage. And then if there is an event of speciation, the graph will divide into two different species. Two extreme, two population will separate out. There is a group of scholar students, there is a group of poor students, and both the groups, they do not interact. They will live separately, and slowly they both will separate in different classes. This is speciation event. This is diversifying or disruptive selection. All right. So see this diagram. Original population. In diagram A, the peak becomes taller and narrower. Then it is stabilizing. If the peak shift occurs towards left or right, then it is directional. And if the entire graph is broken into two different populations, then it is disruptive or diversifying selection. So these are the results of evolution. So types of evolution: convergent and divergent. Results of evolution: stabilizing, directional, disrupting. Remember all these points, all right? And then there is anthropogenic evolution, man-made evolution, for example, industrial melanism, tube-rail mosquito, DDT-resistant mosquito, domesticated cattle. And now, in speciation, we discussed everything, but the word speciation is connected with evolution, but we don't have that much time to discuss types of speciation. But remember, 4 types of speciation: allopatric, sympatric, parapatric, and peripatric speciation. That is important point, but it requires the entire 1 hour session on that particular word. And along with evolution, we should also understand human evolutionary trends. Okay, so these are human evolution. So why we have been different than our ancestor apes? Because of all these changes that have occurred in humans, right? Arboreal to terrestrial life, we lost the opposable toes from the hind limbs. We have opposable toes here, the toes that can touch to all 4 fingers, but our foot toes, they don't touch all 4 fingers. So that is one sign of evolution. Erect posture, bipedalism. There is development of chin, which apes don't have. There is larger size of brain, around 1400 cubic centimeter brain we have. There is development of intelligence and disappearance of simian shell from our jaw, that is mandible. Mandible ની અંદર એક bone છે, simian shell. We don't have that. Therefore, we can, our tongue can move easily in our oral cavity. That's why we can talk. We can have speech. And we use 4 limbs for non-locomotory purpose. So these are the trends of human evolution, but I will skip these slides and I'll directly come to because it's already 5:30, and if you wish to ask me some question, we'll discuss this Hardy-Weinberg principle, founder effect. So many things are there in this particular topic, but asking question is a good evolution, as Darwin says. So if someone has to ask anything, you can. If anyone wishes, you can. See, this is tube-rail mosquito. Because of the lack of the light. This is anthropogenic evolution, right? Any question from any student? Now the session is open for any discussion. The session is open for the next expert speaker also.
Sir, I can unmute them all for. I can give them privileges to unmute themselves for 2 minutes, so they can
Definitely.
ask questions.
Definitely.
Students, you can ask question. You have the privilege to unmute yourself and ask questions from sir. You have 2 minutes.
Okay, in a limited time, I tried to cover so many points in online manner. If I could, make you understand more than 50% of this discussion, this is really successful session, as I believe. Madam, it looks that students hesitate to ask, or they may not have question, or they cannot formulate questions.
Sir, I think we'll leave it to them to ask questions
Yes.
in the group if it is convenient to them that way.
कोई बात नहीं, no problem.
Okay. Thank you so much, sir, for your time and your expert talk.
It was always my pleasure. And the day when this entire activity started, I am taking lectures since then in this Gsbtm workshop in offline, in online manner. So it's always been pleasure to associate with the organization where I work for around 17 years. So thank you to Atmiya University. Thank you to organizer, department, Shivani madam, Praveen sir. Thank you to you, madam. And thanks to all my dear students. Thank you.
Thank you, sir. Okay, students, the next session will be taken by Dr. Deepak Dave. Sir is Associate Professor at Sri M.N. Virani Science College, Department of Physics. Sir has a teaching experience of over 20 years and he'll be teaching you about the basic concepts of electricity. Another topic which is very essential for your competitive exams. And being a biologist, I think most of us fear physics the most. So I think sir has this talent to explain things in a very layman terms, and it would be really beneficial for you if you concentrate. This is the last session for the day. uh, 5 minutes prior to the ending of the session, I'll share the feedback link, and the mock test link will be shared after that, right? Thank you. So I'll ask Dr. Dave to kindly start with his lecture. Thank you, sir.
Okay. Yes, student, I hope so. I may be audible enough. What I have to go through is concepts of current electricity. Now, prior to starting this talk, most important thing about this is, particularly for bioscience student, it is hard to deal with physical sciences and hardcore maths and physics concept. So I used to have lots of chalk and talk work when we are having Gsbtm session up to last year. But this time, due to this unfortunate scenario of COVID, we cannot have physical sessions. So we have to cope up with this online. Okay. So in that case, what I insist, some in between line concept whenever I discuss, all of you make sure that you are noting out it properly. Like slide may have certain limitations. If I'll give formula, but for how to apply that, for that, whenever that kind of concept will come, I'll let you know just to now note down something. That is one. And second, rather than approaching this topic in a theoretical manner, better you try to approach this in a problem solving manner. Okay. So will not have much interaction to do, but still I'll suggest something or some exercise with you people, and I hope so at your end you will try to solve it properly. So at the end of session, when there will be question answer time, you can discuss whether you are having answer or whether you are struggling or not, like that. Okay. So let's start with current electricity. Yes. When we say it is electrical circuit, circuit is a path of different electronic component. If we are having a closed circuit, means there is complete loop of current, complete path of charge to flow, or complete loop of current to flow. Open circuit, naturally it will not allow complete path of electron to flow. Like if you are having any circuit, in between if you are keeping a simple key, when you open the key, circuit will break, so it will become open circuit. So in general, electrical circuit will be path or combination of different electronic component, or sorry, I'll correct myself, electrical component. When it comes to component, okay, it can be a resistor, it can be capacitor, or any other component. Okay, yeah. Now, what electrical current stand for? Okay, now this is first point. Whatever I am displaying, it is mandatory to display in this form, but application is different, so that also I'll differ. First, electrical current is charge in motion. Okay. Now, second point, look, when we say current in metallic conductor, it is created by flow of free electron. because you know that by virtue of its structure, all metallic conductor contain free electrons. Right from valency 1 plus 1 to plus 3. So in case of metallic conductor, current will be due to free electrons, while in case of electrolyte solution, current will be due to ions, that is positive and negative ion. And that is definition of electrolyte you may have gone through during your basic chemistry study. And in case of semiconductor, certain material having conductivity in between, that is more than insulator and less than pure metal. These are semiconductor. Two elemental semiconductor, what we are having is silicon and germanium. So this kind of material having conductivity better than non-metals but inferior than metal. Okay. Now how you may have question from this topic. All three are categorized as conductor. Metal, electrolyte and semiconductor. All are conducting electricity. But out of that, metal are much superior conductor. Two reasons. One is, as metal having conductivity due to free electrons, and electron due to their lighter weight having higher mobility. At the same time, number density of electron. When I say number density, it is number of free electrons per unit volume. In case of metal, is much larger than the number density of ions. In electrolyte, or even number density of electron and holes in case of semiconductor. And conductivity depends on the number density of the charge carrier, as well as it depends on free electron, it depends on mobility, and both is better in case of metal. Okay, prior to starting, let me just reconfirm with our coordinator. Madam, is it audible properly? Yeah, thank you. Yes. As we have discussed this concept, just I have, but for better visibility, we have changed that. Yes. Now, electrical current is amount of charge flowing per time. I is equal to Q by T. Now, this is very primitive concept you have gone through in your secondary schooling. Now, how you may have question from this is, they may ask you to find out number of electrons flowing. By finding or by giving current, they may ask you to find out number of electrons flowing. For that, what concept you are supposed to prepare? Look, I am keeping my cursor on that charge point. I is equal to Q by T. Now this Q, total amount of charge, can also be written. Yeah, I hope so. Now audibility might be proper. Yes, I is equal to Q by T. Now Q, that represents total amount of charge. And total amount of charge is number of electrons into E, N into E, law of quantization. So by giving current and by giving time, they may ask you to find out number of electrons flowing, N. Because Q is equal to IT, then at the same time, Q is equal to N into E. So they may ask you to find out number of free electrons flowing through the conductor. When I say E, it is basic electrical charge. You might be knowing the value 1.6 into 10 raised to minus 19 coulomb. SI unit of charge is ampere. At the same time, remember the CGS unit, it is state ampere or ESU per second. What I have mentioned on the slide is SI unit of charge, that is ampere. In CGS unit, it is state ampere or ESU per second. Ampere is equivalent to coulomb per second, while state ampere is equivalent to ESU, electrostatic unit per second. So naturally, coulomb is SI unit of charge, while ESU, electrostatic unit, is CGS unit of charge. Coulomb represents SI unit of charge and ESU represents CGS unit of charge. At the same time, better all of you note down the value when it comes to relation between coulomb and ESU. 1 coulomb is equal to 3 into 10 raised to 8, 10 raised to 9, correction, ESU. 1 coulomb is equal to 3 into 10 raised to 9 ESU, electrostatic unit. One more unit of electrical charge is also there, that is EMU, electromagnetic unit. And please note down that conversion also. EMU, electromagnetic unit. 1 EMU is 10 ESU. I hope so. All of you might be noting down the important point. Otherwise, in physics, slide will not help all of you much, because after all, it is problem solving subject. Yeah. Number of electrons which are entering and leaving the wires are same, because charge have to be conserved. When current flow through any conductor, naturally no electron is going to create or get destroyed inside the conductor, or even any other charge, like let's say ions in the electrolyte, or holes and electrons in semiconductor. So total charge have to be conserved. So whatever charge entering, same amount of charge leaving, here I have written for electron. Okay. So net zero, yes. Now direction of conventional current. Electron being a negative charge experience force opposite to the direction of electrical field. Student, whatever statement I am giving, that is basic current electricity. It is not part of current electricity, it is electrostatic. But how we are applying that over here? Again, I do repeat. Electron being negative charge experience force opposite to the direction of electrical field. That's why the electrical field, as it is from positive to negative, that's why electron experience force from negative to positive. And the direction of conventional current, I have written current is actually flowing, you have to take it as direction of conventional current is in the direction of flow of positive charge or opposite to the direction of flow of negative charge. Direction of conventional current is in the direction of flow of positive charge or opposite to the direction of flow of negative charge. You may have noted down this point. Yes. Further, when current is remaining constant with time, it is DC. And when current is varying with time, it is AC. Now for DC and AC, looking to the space limitation, I have not gone through much detail, but remember, in DC, whatever value we are mentioning, like 5 ampere DC, it is its absolute value, 5 ampere. But as far as AC is concerned, whatever value of current we are mentioning, it is in terms of its RMS, that is root mean square value. I do repeat, in case of DC, whatever value have been mentioned, that is the value of the current. But in case of AC, whatever value have been mentioned, that is RMS, root mean square value. Now look, why we do require root mean square value? DC, that's okay, it is not changing with time. And it have only one kind of polarity. But AC is periodically changing between two extreme, positive extreme to negative extreme. Let's say plus 5 ampere to minus 5 ampere. So instant to instant value of AC is changing. So one possibility, let's take average. But problem with taking average is, when we take average of two equal and opposite extreme, it will reduce to 0. Okay, like upper side it is going to for plus 5 and lower side it is minus 5. So directly if I'll take average, it will reduce to 0. Yes. One more thing is, if average is reducing to 0, then second possibility is take peak value. But AC is not permanently sustaining over this peak value. It is changing. So first we'll take square. So all square term will be positive. Then take mean and then root. This is pure mathematical process, or you can say statistical process, to find out values of all these physical quantity, not only limited to current electricity. All the physical quantities which are periodically and harmonically changing. When we want to measure value of such quantity, it should always be in terms of root mean square value, RMS. And one more point you are supposed to note down is, if peak value of AC or amplitude of AC is Im, then its RMS value will be peak value by √2. I do repeat, the RMS value of AC signal, whether it is AC current signal or AC voltage signal, it will be peak value by √2. IRMS will be Im by √2. Similarly, VRMS will be Vm by √2. Student, when I say IRMS, I mean by RMS value of current. When I say VRMS, I mean by RMS value of voltage. And look, sometimes examiners are smart enough to give question from this simple topic. Like if I am having 5 ampere AC and 5 ampere DC, then remember 5 ampere AC will be more hazardous than 5 ampere DC. Why? Because when I say 5 ampere DC, it is purely 5 ampere DC. But when I say 5 ampere AC, means that 5 ampere is representing the RMS value. So if RMS value is 5, then I already given relation that RMS is peak by √2. So IRMS is Im by √2. So Im will be √2 into IRMS. So that will be around 7 point something ampere. So if I say 5 ampere AC, then it have capacity to go peak value up to around 7 ampere. So its actual peak value is 7 ampere. But when I say 5 ampere AC, it is 5 ampere DC, it is purely 5 ampere. So naturally, for same magnitude of AC and DC, corresponding AC current will be more hazardous than DC. That was conceptual portion. But at the same time, all of you are supposed to note down this point. AC is always expressed in terms of its RMS value, and RMS value of AC is peak value by √2. I hope so you may have note down this. Yeah. Once we are having idea about current and current electricity, what is reason for current to flow? So right from your secondary schooling, now you are almost on the verge of completion of graduation, but right from your secondary schooling, when the current electricity might have started first time, you may have this concept, only condition current can flow through any conductor is there should be a potential difference. Look, this concept is equally applicable to thermal conductivity as well as electrical conductivity. In case of thermal conductivity through any solid, condition is there should be a temperature difference between two ends of the solids. Same way, in case of electrical conductivity, there should be a potential difference. And the device which gives this potential difference is voltage source. Like a battery or even plug, what you are using is a kind of voltage source. Okay? It can be a chemical cell which is producing DC, direct current, sorry, and generator, electrical generator, which may produce both AC as well as DC. Look, as far as chemical cell is concerned, it is purely developing DC energy. But as far as electrical generator is concerned, it may result in production of AC as well as DC. It depends on the ring what we are attaching at the end of generator. If it is slip ring, it will produce AC. And if it is split ring, it will produce DC. And as I have mentioned the working principle, voltage source, if it is chemical cell, like what you are using in your mobile battery, it is converting chemical energy into electrical energy, while generator is converting mechanical energy into chemical energy. Okay? And in some question, voltage may also be written as EMF, electromotive force. But look, student, this word is little bit misleading. Electromotive force, but make sure that it is not force. It is basically potential difference. The potential difference is a reason which makes charge to flow. Potential difference is reason which makes current or charge to flow. So it provides necessary energy for charge to flow. That's why it is called electromotive force, EMF. But EMF is a voltage, should be expressed in volt. Okay, it is not force which is expressed in Newton. One more thing, from this voltage concept, what you are supposed to write or you are supposed to note down is, when any charge Q pass through potential difference of B volt, then what energy it gain is Q into V. When any charge Q pass through potential difference of B volt, it gain energy is equal to Q into V. This energy may be in terms of kinetic energy or maybe in terms of potential energy. But energy gained by charge will be equal to Q into V, where V is accelerating potential and Q is charge. And student, in particular this point, Q is in energy is equal to Q into V is important in lots of sense, right from de Broglie formula of wavelength of electron, which may you learn in modern physics, to current electricity, even to magnetism. In one or other way, you are supposed to apply this. When any charge Q pass through potential difference of V volt, energy what it gain is Q into V. It can be in terms of kinetic and potential energy. Okay, let me make it clear when it terms of kinetic energy and when it is in terms of potential energy. Student, whatever I am giving you people note, it is for positive charge. So if negative charge question come, then convention will be opposite. What is convention for positive charge? When charge move from higher potential to lower potential, it gain kinetic energy or loss potential energy. Whenever a positive charge is moving from higher potential to lower potential, it is gaining kinetic energy or losing potential energy. And from lower to higher, it gain potential energy and loss kinetic energy. Student, whatever convention I have given, that is purely for positive charge. For negative charge, entire conventions will be reversed. And believe me, this formula you are going to use in lots of different concepts of physics. So prepare it properly. And even 1 electron volt, yes, 1 electron volt definition also come from this. What I said, 1 electron volt, you may have idea, that is unit of energy, which is 1.6 into 10 raised to minus 19 joule. That concept also come from this Q into V. Look how. Take V equal to 1 volt and replace Q with E, that is charge of electron. Then 1 into E, that will give 1.6 into 10 raised to minus 19 joule. And this much energy is 1 electron volt energy. So 1 electron volt energy is change in the energy of electron. I am using the word change because it might be gain or loss of kinetic energy. So change in the kinetic energy of electron while passing through potential difference of 1 volt. That much energy is called 1 electron volt energy. And this conversion is also much useful, particularly for modern physics numericals like dual nature and de Broglie concept numerical. Yeah. One more thing, Ohm's law. Here only in equation form it have been given, but we will discuss in both equation as well as graphical form. Current flowing through any conductor is directly proportional to potential difference applied between two ends of the conductor. Very primitive concept. I is directly proportional to V. Now, how you can have problem from this? So first of all, very basic concept of graph theory. If I plot graph of I versus V, my y coordinate is current and x coordinate is potential. Now, whenever y coordinate is directly proportional to x coordinate, then graph of y versus x will be linear. Okay. So here plot of current versus voltage will be linear or straight line passing through origin. Plot of I versus V graph will be linear passing through origin. And slope of I versus V graph will give inverse of resistance. Do remember, sometime this graph, if they are giving in terms of V versus I, then slope gives resistance. And if it is I versus V, it gives inverse of resistance. So you may have question like two different graphs with different slopes are given. Like I versus V graph you are having, then graph which having higher slope have smaller resistance, and graph having smaller slope having higher resistance. Why? Because here slope is giving 1 by R. So higher the slope, then higher is 1 by R. Higher the 1 by R, then smaller is R. Do prepare it properly. One more thing, what have been written on the slide is current is voltage by resistance. So resistance is equal to current by voltage. Okay. Now think in that way. I have written resistance is equal to current by voltage. So you may have temptation or that kind of thought process. If voltage change or current change, then resistance change. Like when I am writing R is equal to V by I. Resistance of a conductor R is equal to V by I. Now V get double. Then remember, although V get double, then also resistance will not get double, because for any conductor, ratio of potential difference applied to the conductor and current flowing through it remains constant, and that constant is resistance. So V by I is constant, and that constant is resistance of the conductor R. An interesting concept I'll give you from this. Resistance, although I have to only deal with current electricity, but remember, resistance, capacitor, and third is inductor. These are geometrical component rather than electrical component. Look, what I say, it is geometrical component in the sense they are depending on geometry. Later on, I'll discuss how it depends on geometrical parameter like length, area of cross section, type of material. So three basic inherent component. When I say inherent component, means effect of these components are by default present in the circuit. Like wherever I am sitting in one lab, this lab may have wiring and this wiring may have some resistance. Whether I am connecting separate resistance or not, this wire have some resistance. Same way, we can have inductive and capacitive effect. So these three are inherent component of this circuit. Which three inherent component? Resistance, inductor, and capacitor. Their effect are by default present in the circuit. You people belongs to biosciences, so you don't have to go in much deal of that. Otherwise, at high frequency and low frequency, it affects the circuit operation. And all three are geometrical parameter. Geometrical parameter means they are depending on parameters like potential difference, current, and charge, and independent of electrical parameter like voltage. Sorry, I'll correct myself. It was fumble totally. They are independent of parameters like voltage, current, and charge, because they are not electrical parameter. Yes, but they are depending on geometrical parameter like length, area of cross section, and type of material. I hope so. You may have noted down this. Yes. Resistance definition, we are not going for much detail of that, because you may not have question of what resistance of wire. But remember, one concept is SI unit of resistance is ohm. That is not concept. Resistance of even those who have learned up to 10 might be knowing. Even then, they have leave even science also. But remember, when we are using Ohm's law, I versus V graph should be straight line passing through origin. So those devices which are having I versus V graph linear straight line passing through origin are called ohmic devices. Like simple metallic wire is always ohmic in nature, because for metal wire, current is directly proportional to potential difference applied. So it is ohmic. There are certain non-ohmic devices also, like if you are following any semiconductor device, then they are non-ohmic devices. And for non-ohmic devices, IV graph is not always required to be linear. One more thing, in case of ohmic devices, IV relations are 1-to-1. IV relations are 1-to-1 in the sense, for every single value of I, there is correspondingly single value of V. But in non-ohmic, it is not always required. We may have IV relation 1-to-many. So classification you are supposed to prepare, because when any student is appearing for PG exam based on biosciences, physics will always be an elementary level. So remember, ohmic devices are those where IV relations are linear. When I say IV relations are linear, means I versus V graph is straight line passing through origin. For non-ohmic devices, IV relations are non-linear. Yeah. Look, in general, resistance can be of two types. Carbon resistance, which I have displayed on the screen, and wire bond resistance. For carbon resistance, you may have certain question for competitive exam. Carbon resistance are generally very small in size. That's why it is not possible to mention the value of the resistance properly on its body. Although what you are observing on the screen appear much larger, but practically they are just food grain size. So on such small body, it is not much convenient to write down its entire value properly in ohm or kilo ohm. So this resistance or value of these resistors are indicated by color coding scheme, which have been given. The band or the body of the resistor may have 3 or 4 color band. Band corresponding to first is unit position. Like if I'll represent to a number in a mixed manner, mantissa in exponent form. Mantissa means integer and then exponent form. And mantissa, that is a number, digit number. If I am giving in a 2 digit, then first will give unit position. Like red belongs to 1. So here I have given first band, so first I'll write down 1. Then violet, if second, let's say blue is second, it appear like blue, or violet if you are thinking. Having, let's say, band 7, so it is 17, 1, 7. Now whatever the third color band, that you will put as a power of 10. Here third color is yellow. Now digit for yellow is 3. So it is 17 into 10 raised to 3. Fourth color may be there or may not be there. If fourth color is absent, then tolerance is 20%. And if fourth color is golden, tolerance is 10%. If fourth color is silver, it is 5%. Sorry, I'll correct myself. Fourth color is golden, tolerance is 5%. Fourth color absent, it is 20%. And in case of silver, it is 10%. Now, what tolerance means? Tolerance represents the variation of the resistive value in compared to measured value. Like if I am giving resistance of 1 kilo ohm with tolerance of 5% or tolerance of 5%. 5% of 1 kilo ohm, that is 1000, is 50. So resistance might be in the range of 950 to 1050. What value I have given? 1 kilo ohm, that is 1000 ohm. 1000 ohm with tolerance value of 5%. 5% of the original value was 1000, 5%, 50. So value might be vary on lower side 1000 minus 50 and on upper side 1000 plus 50. So it might range in the 950 to 1050. So naturally from it is clear that smaller the tolerance, more the accuracy in the resistance value. Prepare this properly. Yes, during this I have given different digit, like red I said 2, yellow I said 4, tolerance. Now this value you might get in any +2 book or even any basic electronic book. And in that, right from black, brown, red, orange, yellow, green, violet, gray, different colors might be given and which value. And one or other sentence memory tips also different electronics book are giving. Like in our second year BSc student, we are going through, or sorry, first year BSc student, we are going through sentence like B. Roy going to Bombay via Gateway of India. Means black, brown, red, orange, yellow. Means for name, you will take all the alphabet. Then only first alphabet of spelling, going to G, green, Bombay, B, blue, via, violet, gate, way of India, gray, like that. But these kind of sentences you will find in all basic electronics book. Now, this is important point for your application point of view. First, I'll go through this slide and then we'll go through previous. Resistance depends on length as well as area of cross section. Resistance of wire is directly proportional to length and inversely proportional to area of the cross section. Larger the length, more will be resistance. Larger the area, smaller will be resistance. Okay. Now look, resistance is directly proportional to length, but in some question you may get answer by taking resistance proportional to square of length rather than resistance proportional to length. Like if I am stretching the wire and length of wire become double, what is its new resistance? Then if you are following this formula R proportional to rho L by A, you will get that when length wire is stretched, its length become double, resistance become double. And student, paper setter are smart enough to put such options. They will put double, but answer is 4 times. Look, the logic is whenever we are stretching the wire, then length as well as area of cross section of the wire both are changing. That's why if length become double, area will reduce to half. So resistance become 4 times. So in general, what you are supposed to prepare, if I am comparing resistance of two separate wires, or even if I am cutting the wire, so in that case, you will follow the proportionality that R proportional to L. Yes, because whenever you are cutting the wire, area of cross section is not getting changed. That's why you will take R proportional to L. Even you are comparing resistance of two separate wires having same area of cross section, then also you can take R proportional to L. But if you are stretching the wire or if you are melting and remolding the wire, in both cases, either stretching the wire or melting and remolding the wire, both parameters will get affected, length as well as area of cross section. So in this kind of question, you have to take R proportional to L square. Now for that 3 points, I am giving those students who want can note down, and it is important for lots of problem solving. Point number 1, when wire is stretched so that length of wire increase by n time, then its resistance will become n square time. Again, I do repeat, when wire is stretched so that length of wire increase to n time, then resistance become n square time. Something similar, when wire is stretched so that area of cross section of wire reduced to 1 by n time, in that case also resistance will become n square time, because conceptually both things are same. Either you say length increased to n time or area reduced to 1 by n time, both are same, because total volume have to be remain constant and volume is product of area and length. So if one is increasing by n time, second should reduce by n time. So either you say wire is uniformly stretched so that length of wire increased to n time, or area reduced to 1 by n time. In both cases, resistance of wire become n square time. Yes. One more concept, when wire is uniformly stretched so that radius, length we have already discussed. I'll go through radius of cross section. Wire is uniformly stretched so that the radius of cross section of wire reduced to 1 by n time. In that case, the resistance will become n raised to 4 time. Why? Because radius is reducing to 1 by n time, then area will reduce to 1 by n square time, because area pi r square. So effect will be raised to 4. So when a wire is uniformly stretched, radius reduced to 1 by n time, resistance become n raised to 4 time. I hope so all of you may have noted down this point while cutting or comparing resistance of two separate wires. The proportionality what we'll take is R proportional to L. But while stretching the wire or melting and remolding the wire, Proportionality, what you will take is R proportional to L square. And remaining consequence I have discussed how area and radius are affecting wire. Yes. Up to this, or this concept was how resistance is depending on the geometrical parameters like area and length. At the same time, resistance is also temperature dependence. Resistance at certain reference temperature T1 is R1, then resistance at certain temperature T is R2, then we can take R2 is equal to R1 inside the bracket 1 plus alpha T2 minus T1. Here R1 is resistance at some reference temperature, R2 is resistance at some higher temperature. Okay. Student, as far as reference temperature is concerned, generally 0 degree, 20 degree and 27 degree. Resistance at 0 degree is available, resistance at 27 degrees Celsius is available, or resistance at 20 degrees Celsius is available, then that we can take as a reference temperature. But otherwise, don't mistake that all lower temperature are not reference temperature. You cannot apply like resistance at 60 degrees Celsius is available and resistance at 100 degrees to be find out. Then resistance at 60 degrees is not reference temperature. You have to use separately for 60 and 100, and then you can take ratio to get answer. So remember here R1 is resistance at some lower temperature, but that lower temperature should be reference temperature 0, 20 or 27. R2 is resistance at certain high temperature. Alpha is temperature coefficient of resistance or temperature coefficient of resistivity. Alpha in case of metallic conductor, alpha is positive. That's why for metallic conductor, as temperature increase, its resistance increases. As temperature increase, for metallic conductor resistance increases. In case of semiconducting material, alpha is negative. Temperature coefficient of resistivity for semiconductor is negative. That's why for semiconducting material, as temperature increase, resistivity decrease. And here student, there is third classification for certain alloys like tungsten or copper consonant. Their alpha is negligibly small. It is much smaller than for metals or for semiconductor. Although metal have positive alpha, semiconductor have negative alpha. So if we are comparing with metal, for alpha for alloys like tungsten or copper consonant is very small. That's why for such alloy, resistivity will almost remains constant with temperature. Resistivity of alloy is almost remaining constant with temperature because it's smaller value of alpha. And that's why alloys of such wires of such alloys are used to make standard resistor called wire wound resistor. Binding of such wire will make standard resistor. Standard resistors in the sense they are not much varying with temperature. But problem at the same time with such resistors are they are very bulky in size. So in practice, what we are using is carbon resistor, which I already discussed. Student, this formula you are supposed to prepare properly. R is equal to R1 into 1 plus alpha T2 minus T1. In application point of view, we have discussed this concept that in case of R1, that is resistance at reference temperature, all lower temperature you will not have to take reference temperature. Generally, which temperatures you can take reference temperature, we have already discussed. Positive and negative alpha have also been discussed. Yes, combination of resistors. Look, out of series and parallel for understanding purpose, one figure I have given. But this concept you have to prepare in an entire different manner. Equivalent resistance for series, nobody will have much issue with that. R is equal to R1 plus up to so on. Parallel 1 by R is equal to 1 by R1 plus up to so on. Okay. In addition to that, what you are supposed to prepare. Look, as far as figure is concerned, I have given you parallel, and concept I have given series. Why? So both things can be covered in a given time part. So look, here, this one, for parallel combination, different resistors are in parallel. Remember, potential through all the resistors will be same. When two or more than two components are in parallel, the potential difference will remain same. And current will be divided. And current will be divided with inverse proportion of resistance. Like here I1, I2, I3 given. Let we are having two resistors I1 and I2. And total amount of current I have to be divided. Then what will be current through first I1? Then I1 will be total current I into R2. If we are finding current through resistor R1, then it will be I into R2 upon R1 plus R2. Same way, if we are finding current in R2, then I2 will be I into R1 upon R1 plus R2. Now, how you may have question from this in a problem solving method, like total amount of current I have to be divided into two resistors of 4 and 5 ohm in parallel. Then what will be ratio of I1 is to I2? So I1 is to I2 will be R2 is to R1. So it will be 5 is to 4. Smaller the resistance, larger will be current. So prepare that kind of question properly. Yes. Another concept from current electricity you are supposed to prepare is power. Now for electrical power, yes, one formula have been displayed on the board or displayed on the screen, but three you are supposed to prepare at the same time. Which one you have to apply? Power, what I have displayed is current into voltage. P is equal to I into V. But as per Ohm's law, V I can replace with I by R. Now, if I am replacing V with I by R, then same formula will change to I square into R. At the same time, if I am replacing I is equal to V by R, then this formula will reduce to V square by R. So, electrical power I square R, V square by R, or V into I. V into I have been displayed on the screen. Now, concept is, if you are following electrical power P is equal to I square into R, then what you are getting? Power consumed is directly proportional to resistance. If you are using electrical power is equal to V square by R, then what you are getting? Power consumed is inversely proportional to resistance. Okay? And if you are taking P is equal to V into I, then what you are getting? Power consumption have nothing to do with resistance. It is independent of resistance. So student, same concept, we are having three different condition or three different answer. One, if I am using P is equal to I square R, power consumption is directly proportional to resistance. If I am using P is equal to V square by R, then power consumption is inversely proportional to resistance. And if I am using P is equal to V into I, then power consumption is independent of resistance. Then naturally answer will differ, which means there should be some situation in which we can apply which formula. First, I square R. I square R concept you have to use in which case? Whenever different components are connected in series and you are finding power consumption among them. Like two resistors are in series, 5 ohm and 10 ohm. We are supposed to find out power consumption in both. So naturally power consumption in 10 will be double than power consumption in 5. Why? Because as both are in series, so formula power we can use as I square into R. In contrast to that, if I am having both resistors are in parallel, then power consumption will be double in that 5 ohm resistor rather than 10 ohm. Why power consumption will be double in 5 ohm resistor? Because as both are in parallel, so rather than using I square R, the concept or formula what we are supposed to use is V square by R. So student, same situation, two different answer. First, higher the resistance, more is power consumption. Second,
smaller the resistance, more is the power consumption. Because in first we have used I square R, in second we have used V square by R. So how to decide whether to use I square R or V square by R? Condition I have given. In series combination, you will use I square R. In parallel combination, you will use V square by R. Third condition, V into I. Now remember, V into I you are never supposed to use for power consumption. Although power is V into I have been displayed on the screen, but V into I you will never use for power consumption. So in which situation we have to use V into I? V into I gives energy available from source. V into I gives energy available from source. Like if I am having a chemical cell of voltage rating of 10 volt and current rating of 1 ampere, then its power rating will be 10 watt. In that case, I have to use V into I. V into I is exclusively you are supposed to use for power available from source. V into I you never are you never have to use to find out any sort of power consumption. When to use I square R and when to use V square by R, that we have made it clear. Yeah. Kilo conversion is quite whatever. Whenever kilo prefix is there, it is 10 raised to 3. You people are almost graduate student. You are so not supposed to go through that. Yes. One formula or concept have been asked to you. Yes, one more thing. Yes, for power, prepare this concept 1 kWh power, that is 1 unit of power. What electrical power consumption is given in unit, 1 kWh is 1 unit. This conversion also way back in your schooling you may have learned, but if you are not ready with that, then prepare this. It may be useful for lots of questions. 1 unit of power, that is 1 kWh, is 3.6 into 10 raised to 6 joule. So kilowatt hour, it pronounced like unit of power, but actually it is unit of electrical energy, kWh, and 1 kilowatt hour is 3.6 into 10 raised to 6 joule of energy. Student, last concept is different electrical tool, voltmeter, to measure electrical potential difference. And remember, voltmeter in circuit should always be connected in parallel. Like if I want to find out potential difference across any component, voltmeter should always be connected in parallel with that component. One more thing for voltmeter, voltmeter should be device with very high internal resistance. The resistance of voltmeter should be very large, ideally infinite. Resistance of ideal voltmeter should range up to infinite. Ammeter is measuring current and indirectly charge. An ammeter should always be connected in parallel. Sorry, correction, series. Voltmeter in parallel, ammeter in series. And as ammeter is in series, the internal resistance of ammeter should be negligibly small, ideally 0. Ohm meter is used to measure the resistance, and if you are having a multimeter, then remember the ohm meter will have a reverse scale. A reverse scale in the sense like voltmeter and ammeter scale are having deflection from left to right. Then ohm meter scale will have deflection from right to left. When it comes to 0, means it have least resistance and opposite concept. So ohm meter scale will have opposite deflection. Like 0 deflection, 0 volt, 0 current, but in ohm meter, if it is 0 deflection, it is larger resistance. So suppose to prepare these 3 uses also. Now before I conclude, some suggestions. If biology student are preparing for any PG exam where physics is part of their paper, then remember physics will always be very elementary for you people. So as far as possible, rather than going for much theoretical concept and reading much theoretical book, for physics, better you go for problem solving, and problem solving of those exams which are specifically targeted for biological sciences student. Why? Because physics will also differ for gate or net physics and physics concept for biological science, because they will have some application hardcore mathematical concept. You may have only elementary kind of concept to work with, because majority of you may have leave your physics after 12 or after first year. So they are expecting only that much to be prepared by you. Prepare resistance concept, its dependency on geometrical parameter, temperature. Prepare electrical power concept properly, and combination of resistance and division. I hope so this session might help you to make some point clear and to solve problems of current electricity. Now, I'll request coordinator ma'am to make this session open for any question, any doubt. Yeah, ma'am.
Students, the session is open. You have the privilege to unmute yourself and ask questions if you have any.
Yes, whatever, lots of in between line I have discussed, whatever, if you have not down and if you have any question, even you can drop your query to the coordinator also, coordinator of day or coordinator of the workshop, and they will pass same to me, so I can respond you to later on also. But if any of you having any doubt of any query just right now, better we discuss at this instant only. Yeah, student. Okay. Yes, I can understand physics might not be your cup of tea, and immediate response in physics cannot be expected much from biosciences student. But whenever you want, you can have query drops to the coordinator. They will pass, and I will positively respond to you. All the best for all kind of competitive exams and entrance exam you are going to face. Yes, all the best again. And thanks for giving attention for dry subject like physics for biosciences student. Yeah, thank you very much.
Thank you so much, sir. I'm sharing the link for today's feedback. I request you to kindly fill it.
Thanks, ma'am, for your support.
Most welcome.